Hey everyone, I am Dr. Mungli. So in this video, I will be going over entire metabolism. So this video is suitable for those who have already studied biochemistry and all the metabolic pathways, and also for those who are reviewing for their exam, whatever the type of biochemistry exam it is, including a competitive exam like USMLE Step-1 exam or NEET PG exam or NBME exam if you are taking, or if you are taking exit exam like FMGE exam. So whatever the type of biochemistry exam it is, so this video will be useful to you. So listen to me carefully. I will be going over a little bit faster, and also I will cover all the important enzymes and clinical application of those enzymes in brief. So all the important stuff, every enzyme that is important in the metabolic pathways, entire thing, as you see here, almost every important aspect of metabolism I have written here. So please be with me. We will finish this all hopefully in 1.5 to 2 hours, probably little less than that, or maybe little more than that. Let's get into that. So first I will go with the carbohydrate metabolism, and then I will connect that with the lipid metabolism, and then I will take you into amino acid metabolism. So as always, carbohydrate metabolism, we will start from glucose. So glucose, which is a hexose sugar having 6 carbon, undergoing glycolysis. Glycolysis, it's an oxidation of glucose. So glucose breaking down all the way into pyruvate. So that means pyruvate is a 3 carbon molecule and glucose is a 6 carbon molecule. So obviously when one glucose is undergoing oxidation in glycolytic step, glycolysis, so there will be formation of 2 pyruvate. Remember that. So one glucose breaks into 2 pyruvate molecules. So each pyruvate is a 3 carbon. So 3 plus 3, 6 carbon. So anyway, so there are 3 irreversible reactions in glycolysis that you must remember. Sort all those 3 irreversible reactions. So very first reaction in glycolysis is an irreversible reaction, and that is catalyzed by glucokinase in the liver and beta cells of pancreas and hexokinase in the peripheral tissue. Second irreversible step in glycolysis is conversion of fructose 6-phosphate into fructose 1,6 biphosphate, and that is catalyzed by phosphofructokinase 1 enzyme or PFK1 enzyme. And the third, that's the second irreversible, and the third irreversible step in glycolysis is conversion of phosphoenol pyruvate into pyruvate done by pyruvate kinase enzyme. So these are 3 irreversible steps in glycolysis. One more time, it is hexokinase glucokinase reaction, conversion of glucose into glucose 6-phosphate, and then fructose 6-phosphate into fructose 1,6 bisphosphate done by PFK1, phosphofructokinase number 1, and then phosphoenol pyruvate to pyruvate reaction catalyzed by pyruvate kinase reaction. These are the 3 irreversible reactions that you should remember to know glycolysis. Now the most important step in glycolysis, and also the most regulated and the rate limiting or rate determining step of glycolysis, which is also referred as a master controller of glycolysis, it is phosphofructokinase number 1, and that is conversion of fructose 6-phosphate into fructose 1,6 biphosphate. Anyway, let's get into each one of them. Glucose into glucose 6-phosphate done by hexokinase and glucokinase. Remember, hexokinase there in the peripheral tissue, glucokinase there in the liver and beta cells of pancreas. Hexokinase is a low Km and low Vmax enzyme, whereas glucokinase is a high Km, high Vmax enzyme. So glucokinase being present in the liver, it will make sure that during postprandial condition, blood glucose is brought to the normal range, because glucokinase is a high Km and also it's a high Vmax enzyme. Whereas glucokinase being present in the beta cells of pancreas, it is making sure whenever there is higher glucose levels in the blood, insulin is released from the beta cells of pancreas, because you know insulin is a hypoglycemic hormone. It is trying to bring down glucose level, and so that basically normalizing the blood glucose level under postprandial conditions. Now Conversion of those are the two enzymes there, and also the applied clinical aspect is like if there is a mutation in pancreatic glucokinase enzyme, it will give rise to a disease called MODY type 2, maturity onset of the young type 2, remember that. Now, conversion of glucose 6-phosphate to fructose 6-phosphate, it's a reversible reaction. So, that's the phosphoexose isomerase enzyme. Fructose 6-phosphate into fructose 1,6-bisphosphate, and that's one of the second irreversible reaction in glycolysis done by phosphofructokinase 1 enzyme, and this particular enzyme is regulated by various allosteric regulators. So, two negative allosteric regulators of PFK1 is it is ATP and citrate. ATP, higher ATP levels in the cytoplasm, higher citrate levels in the cytoplasm will have a negative effect on this enzyme, thereby decreasing the pace of glycolysis. And whenever there is higher levels of AMP, that is adenosine monophosphate, and whenever there is higher levels of fructose 2,6-bisphosphate, and these molecules, they will act as a positive allosteric modulators on PFK1 enzyme. So, especially in the liver, out of all, that is AMP, ATP and citrate, so fructose 2,6-bisphosphate is the major regulator of PFK1 in the liver, because higher the levels of fructose 2,6-bisphosphate in the liver will keep PFK1 active, most active, and keep glycolysis active. And whenever there is a lower levels of fructose 2,6-bisphosphate, glycolysis kind of slows down because its positive effect on PFK1 is less. So, concentration of fructose 2,6-bisphosphate, it is maintained by two hormones. One is insulin will increase fructose 2,6-bisphosphate concentration, whereas glucagon will decrease fructose 2,6-bisphosphate concentration, and that will be done by these two enzymes by acting on another enzyme called PFK2, that is phosphofructokinase 2, which is a bifunctional enzyme. It has two subunits, catalytic subunit. One is a kinase subunit, other is phosphatase subunit. So, whenever kinase subunit of PFK2 is high or active, at that time more and more fructose 2,6-bisphosphate is synthesized, and thereby higher glycolysis is going on, and this is promoted by insulin by keeping PFK2 in dephosphorylated condition. Whereas glucagon under fasting condition, what it does, it is going to keep PFK2 phosphorylated, thereby it is going to activate phosphatase subunit of PFK2, thereby it decreases the concentration of fructose 2,6-bisphosphate, thereby its positive effect on PFK1 is not there. So, this is what is the regulation there. And also glucokinase and hexokinase regulation. So, hexokinase is feedback inhibited by its immediate end product glucose 6-phosphate, whereas similar feedback inhibition is not there over glucokinase. And note that glucokinase in the liver is induced by insulin, whereas similar induction by insulin is not there over hexokinase, remember that. Now, coming to conversion of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So, this will be done by enzyme aldolase B. That aldolase B enzyme is a lyase class of enzyme. It will break a 6-carbon fructose 1,6-bisphosphate into 3-carbon glyceraldehyde 3-phosphate and 3-carbon dihydroxyacetone phosphate. There are 3 aldolases, aldolase A, B and C. Aldolase B is there in the liver. So, other aldolases like aldolase A, aldolase C, it is there in other tissues. Now, glyceraldehyde 3-phosphate and it is a reversible reaction. Glyceraldehyde 3-phosphate can run down into glycolysis all the way to pyruvate, whereas dihydroxyacetone phosphate, depending on the tissues, depending on the metabolic conditions of the tissue, dihydroxyacetone phosphate either it can be converted back into glyceraldehyde 3-phosphate done by triose phosphate isomerase enzyme, or dihydroxyacetone phosphate can be converted into glycerol 3-phosphate, and that will be done by enzyme glycerol 3-phosphate dehydrogenase. Okay, and that glycerol 3-phosphate, especially in the liver and tissues wherever esterification is going on. So, glycerol 3-phosphate, it is going to esterify fatty acid to make triacyl glycerol, and that triacyl glycerol, if it is there in the liver, it will be loaded on to very low density lipoprotein. I am going to come to that point later. Anyway, so for our understanding purpose, what we will do, we are going to take dihydroxyacetone phosphate into glyceroldehyde 3-phosphate done by triose phosphate isomerase. And now that means we have 2 glyceroldehyde 3-phosphates now, because one of the dihydroxyacetone phosphate is diverted into glyceroldehyde 3-phosphate, and originally we had 1 glyceroldehyde 3-phosphate from fructose 1,6-bisphosphate. Now these 2 glyceroldehyde 3-phosphates, they will go down into 1,3-bisphosphoglycerate, and that will be done by glyceroldehyde 3-phosphate dehydrogenase enzyme, and that reaction needs inorganic phosphate. So inorganic phosphate will get into this reaction, and also this reaction needs NAD. NAD gets into the reaction, and NADH plus H plus will come out of this reaction. So that means there is a requirement of NAD here in the glycolysis. So that NAD, as you know, it will come from niacin, vitamin B3 niacin. Inorganic phosphate will come from inorganic phosphate pool. Remember, whenever person has got higher levels of arsenate, like arsenate toxicity, at that time this arsenate is going to go and bind to inorganic phosphate binding site. Thereby it will have a negative effect on this particular enzyme. So whenever arsenic is going to, whenever person has got higher levels of arsenic, this arsenic will go and bind to other site, allosteric sites, and thereby it will have a negative effect on this particular reaction. That means arsenic is not going to allow conversion of glyceroldehyde 3-phosphate into 1,3-bisphosphoglycerate. Rather than what it does, it is going to take glyceroldehyde 3-phosphate all the way into 3-phosphoglycerate, and it bypasses this 1,3-bisphosphoglycerate step. Now let's see what happens to this 1,3-bisphosphoglycerate reaction, and then I'll come back to arsenic. So 1,3-bisphosphoglycerate will be converted to 3-phosphoglycerate. During that time, there will be ADP that is getting into the reaction. Adenosine diphosphate gets into the reaction and converted to adenosine triphosphate. So that means there is a formation of ATP in the cytoplasm. Glycolysis, conversion of glucose all the way to pyruvate is going on in the cytoplasm. It's a cytoplasmic pathway. So there is a formation of ATP in the cytoplasm. This type of ATP generation we call it as substrate level phosphorylation, because ADP is phosphorylated with one of the phosphate coming from the first carbon of 1,3-bisphosphoglycerate to make ATP. So there is a phosphorylation and generation of ATP in this particular reaction. That's why this is called as substrate level phosphorylation. One of the example for substrate level phosphorylation is conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate, and this reaction is done by phosphoglycerate kinase enzyme. Remember that. Now, so what happens with the arsenic? Arsenic, what it does, it is going to divert glyceroldehyde 3-phosphate directly into 3-phosphoglycerate. That means a cell don't get that NADH plus, it is not going to make ATP. Note that one NADH plus, it's an energy rich molecule. If it goes to electron transport chain by malate aspartate shuttle mechanism, it's going to give 2.5 ATP. That means we have 2 glyceroldehyde 3-phosphates here. That means we are going to get 2 NADH plus, and also down here we are going to get 2 ATPs, right? So 2 NADH plus is 2.5 plus 2.5, 5 ATPs there and 2 ATP here. For every 2 glyceroldehyde 3-phosphate making 2 3-phosphoglycerate. So that means arsenic, by bypassing glyceroldehyde 3-phosphate all the way into 3-phosphoglycerate, it is starving the cells from lack of energy. That's important mechanism of arsenic on glyceroldehyde 3-phosphate dehydrogenase enzyme. You got to remember that. Now, what happens to the 3-phosphoglycerate? 3-phosphoglycerate converted to 2-phosphoglycerate by phosphoglycerate mutase enzyme. Not really that important. 2-phosphoglycerate to phosphoenol pyruvate is done by enolase enzyme. And what? It's an hydrolase category of enzyme. So enolase is a lyase category of enzyme. So enolase, what it does, it is going to help in the conversion of 2-phosphoglycerate into phosphoenol pyruvate. What is the clinical importance of this enzyme? Enolase is inhibited by fluoride, by non-competitive mechanism. So by fluoride, what it does at certain concentration, especially the amount of fluoride used in the toothpaste or oral anti-cavity solution, it is going to have an inhibitory effect on bacterial enolase enzyme. You note that anaerobic bacteria in the mouth, so they use glycolysis, anaerobic glycolysis to generate ATP. So by using a fluoride in the toothpaste or oral anti-cavity solution, so you are inhibiting bacterial enolase enzyme, thereby going to decrease glycolysis and starve those bacteria of lack of energy, and thereby they don't multiply, and thereby it helps in prevention of dental cavities or tooth decay is prevented by using fluoride. And also note that fluoride is used in blood sample collection. Whenever blood sample is collected for biochemistry lab determination, some of the parameters determination, so like blood glucose determination, so we want to keep this glycolysis down, so that whatever the glucose is there at the time of collection, so we want that glucose to be determined. So we are halting the glycolysis by using fluoride, because you know fluoride inhibits enolase enzyme in the glycolysis. Okay, now that's the clinical aspect of it. Now what happens to the phosphoenol pyruvate? Phosphoenol pyruvate is converted to pyruvate, that is done by pyruvate kinase enzyme. It's the third irreversible step in glycolysis. So in this step again, ADP, that is adenosine diphosphate, getting into the reaction, and ATP comes out of the reaction. That means for every 2 phosphoenol pyruvate making into 2 pyruvate, we are going to get 2 ATPs there. So how many ATPs now? So glucose to glucose 6 phosphate, there is a consumption of 1 ATP. So 1 ATP gets into the reaction and ADP comes out here. So that means there is consumption 1 ATP there. Fructose 6 phosphate into fructose 1 6 bisphosphate, another ATP is consumed. So 2 ATPs are consumed there. Whereas here 2 ATPs are made in phosphoglycerate kinase reaction, and 2 ATPs are made in pyruvate kinase reaction. So 4 ATPs are made. So 4 minus 2, these 2 ATPs are consumed there. So 4 minus 2 ATPs. So to net 2 ATPs we are going to gain from 1 glucose going all the way into 2 pyruvate. And also if you calculate these NADHH plus, because we have 2 NADHH plus, so 2.5 plus 2.5. So that means we are going to get 5 ATP in this step. Using malate aspartate shuttle mechanism. So 5 plus 2, 7 ATPs we are going to get under aerobic condition when 1 glucose is converted into pyruvate. Whenever cell has sufficient oxygen, sufficient like mitochondria is functional, at that time conversion of 1 glucose into 2 pyruvate we are going to get. So under anaerobic condition, so what happens? Pyruvate will be diverted into lactate whenever cell has lack of oxygen and lack of mitochondria. So lactate dehydrogenase enzyme here, what it does, it's going to convert or it's going to reduce pyruvate into lactate. In order to do that, what it does, it is going to take this NADHH plus, NADHH plus comes here and it will be oxidized into NAD, and NAD will go back to the cytoplasm and used in this reaction. That means lactate dehydrogenase enzyme, it helps in regeneration of NAD plus, to continue glycolysis. That's the only reason why pyruvate is diverted into lactate under anaerobic condition. Remember that, regeneration of NAD plus, because we have only limited quantities of NAD plus in the cytoplasm. NADH has to be continuously regenerated into NAD plus. So when the cell has less oxygen and lack of mitochondria, so only way to regenerate that, one of the way to regenerate is to convert pyruvate into lactate and make sure NAD is available and glycolysis is continued. So that's how 1 glucose will be converted to 2 lactate under anaerobic condition. Under aerobic condition, glucose to 2 pyruvate. 2 pyruvates will go into mitochondria if the cell has mitochondria and sufficient oxygen, and it will undergo TCA cycle, all that. I am going to come to that point. Now, so this is what is all about glycolysis, oxidation of glucose into pyruvate, 2 pyruvate, and conversion of pyruvate into lactate under anaerobic condition, and when the cell has when the cell don't have mitochondria, like red blood cell. Okay. So how many ATPs? From 1 glucose to 2 pyruvate we're going to get under aerobic condition, we're going to get net 7 ATPs. And under anaerobic condition, 1 glucose to 2 lactate, we're going to get 2 net ATPs. And any situation which creates anaerobic condition, it may be like red blood cell, it may be some of the tissues where lack of oxygen is there, it may be lens, it may be retina, or it may be like kidney, adrenal, kidney medulla, things like that, or like skin grafting, when the newly grafted skin is there where revascularization yet to take place. During that, and myocardial infarction, whenever like septic shock, these condition when lack of oxygen is there, so that will lead to anaerobic glycolysis. Remember all the scenarios, very important. Okay, that's about glycolysis. Regulation also, and pyruvate kinase enzyme is one of the regulated enzyme. Note that this fructose 1,6 bisphosphate has a feed forward activation on pyruvate kinase enzyme. Remember that, feed forward activation. And the excess ATPs, excess NADH plus molecule, excess acetyl-CoA will have a negative effect on pyruvate kinase enzyme. Even excess long chain fatty acids will have a negative effect on pyruvate kinase enzyme. So this is all about glycolysis down there. Now, before I take you into other metabolic pathways, so let me first explain you what happens to the pyruvate when the cell has mitochondria and sufficient oxygen. So whenever cell has mitochondria and sufficient oxygen, so pyruvate is oxidized into acetyl-CoA. During this process, there will be release of carbon dioxide, because 3 carbon pyruvate will be converted into 2 carbon acetyl-CoA. So there is a decarboxylation going on, and also NADH plus will come out of the reaction. NAD gets in and NADH plus comes out of the reaction. So there is a formation of NADH plus. So this particular reaction is catalyzed by pyruvate dehydrogenase complex, and this is called as a link reaction because it is linking glycolysis with the TCA cycle. Now, what is the specialty of pyruvate dehydrogenase complex? Pyruvate major subunits, E1, E2 and E3 subunit. E1 subunit, what it does, it causes decarboxylation of pyruvate. So that's why it is called as pyruvate decarboxylase subunit. And then E2 subunit, what it does, it is going to take that organic molecule after decarboxylation and attached with a lipoate. So that's why it is called as dihydrolipoyl transacetylase enzyme. And then E3 subunit, what it does, it is going to reduce FAD into FADH plus, and FADH plus later reduce NAD into NADH plus. That's why it is called as dihydrolipoyl dehydrogenase enzyme. So anyway, with E1, E2, E3 subunits, so there is requirement of 5 coenzymes. What are those 5 coenzymes? One is TPP, active form of thiamine, thiamine pyrophosphate, one of the requirement. And then you need lipoate, and then you need pantothenate, which is coenzyme A. Pantothenate derivative is coenzyme A, SH, COSH. And then riboflavin derivative FAD and niacin derivative NAD. So we need 5 coenzymes for optimal functioning of pyruvate dehydrogenase complex, which is converting pyruvate into acetyl-CoA. If there is a mutation in a gene coding for pyruvate dehydrogenase complex, like usually E1 subunit, if it is mutated, that's a commonly mutated subunit. It's an X-linked dominant condition. So it will give rise to what is called as congenital lactic acidosis. Why? Because when pyruvate is not diverted sufficiently into acetyl-CoA, so pyruvate will go into lactate, so there will be more lactate formation. So excess lactate will like build up in the blood, in the tissues, that will give rise to congenital lactic acidosis. That's because of mutation in E1 subunit of pyruvate dehydrogenase complex. Now, there is out of E1, E2, E3 subunits, E1 and E2 subunits, they have common gene for all other 2 dehydrogenase complexes, because not only pyruvate dehydrogenase complex needs E1, E2, E3, other 2 dehydrogenase complexes, that is alpha ketoglutarate dehydrogenase complex in the TCA cycle and branched chain alpha keto acid dehydrogenase complex, which is involved in branched chain amino acid metabolism. So these 3 enzymes, they have similar subunit arrangement, E1, E2 and E3 subunit. So out of these three enzymes, these three subunits, E1 and E2 is coded by separate genes for each one of these three enzymes, whereas E3 subunit gene is shared by all the three enzymes. That means if there is a mutation in E3 subunit, it will lead to decrease in the functioning of pyruvate dehydrogenase complex, leading to elevation of pyruvate, decreasing the function of alpha ketoglutarate dehydrogenase complex here, leading to elevation of alpha ketoglutarate, and decrease in the functioning of this enzyme here, branched chain alpha keto acid dehydrogenase complex, leading to elevation of all these branched chain alpha keto acids. So that means pyruvate is an alpha keto acid, alpha ketoglutarate is an alpha keto acid, and branched chain alpha keto acids here, alpha keto isovalerate, alpha keto beta methyl valerate, alpha keto isocaproate, they are all alpha keto acids. That means if there is a E3 subunit mutation, gene mutated, it will give rise to excess elevation of alpha keto acids, leading to metabolic acidosis. So that's what is the application of here. So now that's about pyruvate dehydrogenase enzyme complex. Coming to regulation of pyruvate dehydrogenase enzyme complex, acetyl-CoA, whenever it accumulates, it will have a negative effect on pyruvate dehydrogenase complex. NADH, whenever it accumulates, it will have a negative effect on pyruvate dehydrogenase complex. And also note that pyruvate dehydrogenase enzyme is regulated by two more associated subunits. One is pyruvate dehydrogenase enzyme kinase, pyruvate dehydrogenase phosphatase. So whenever pyruvate dehydrogenase kinase is active, pyruvate dehydrogenase enzyme complex becomes inactive because it is phosphorylated. Phosphorylated form of an pyruvate dehydrogenase complex is an inactive enzyme. Whereas whenever phosphatase is activated, at that time, so enzyme becomes active. So calcium usually keep pyruvate dehydrogenase complex phosphatase active, thereby keep pyruvate dehydrogenase complex in dephosphorylated state, and that's an active form of an enzyme. And insulin is going to keep pyruvate dehydrogenase complex active, whereas glucagon is going to keep pyruvate dehydrogenase complex inactive. These are all points about link reaction, which is converting pyruvate into acetyl-CoA. Now, what happens to the acetyl-CoA? Acetyl-CoA, it condenses with oxaloacetate. Acetyl-CoA is a 2-carbon molecule. Oxaloacetate is a 4-carbon molecule. So 2-carbon molecule combines with 4-carbon oxaloacetate to make citrate, and citrate or citrate is a 6-carbon molecule. It has got 3 carboxylate groups. That's why this cycle, it is called as citric acid cycle or tricarboxylic acid cycle, also called as Krebs cycle. Okay, now citrate, it is converted into isocitrate, and that is done by aconitase enzyme. And importance of aconitase here is a rat poison fluoroacetate, which is converted into fluorocitrate, and that fluorocitrate will have a non-competitive inhibitory effect on aconitase enzyme here. Conversion of citrate into isocitrate done by aconitase, inhibited by rat poison fluorocitrate, and that's the application there, you should remember. Now isocitrate is converted into alpha ketoglutarate. This will be done by enzyme isocitrate dehydrogenase, and this isocitrate dehydrogenase enzyme is one of the regulated enzyme. In fact, it is one of the most regulated enzyme in TCA cycle. I will come to that point later. And also in this reaction, there will be release of carbon dioxide. So 2-carbon acetyl-CoA is going to release one of the carbon there. So I will write 2 carbons in blue color there. So 2 carbons, one of the carbon is released in this step, isocitrate dehydrogenase step. So that carbon actually it will be coming from acetyl-CoA. Now that means 6 carbon isocitrate becomes 5 carbon, 6 carbon becomes 5 carbon now, 5 carbon alpha ketoglutarate. Now this 5 carbon alpha ketoglutarate, and also in this step isocitrate dehydrogenase step, there will be generation of one NADH plus H plus molecule. Now, so alpha ketoglutarate is undergoing another decarboxylation process here. Carbon dioxide is released, and also there will be release of one more NADH plus H plus molecule. So that will be done by alpha ketoglutarate dehydrogenase complex. I told you this is similar to pyruvate dehydrogenase complex, conversion of pyruvate into acetyl-CoA. And also I forgot to tell you, this pyruvate dehydrogenase complex is inhibited by arsenic. This arsenic will go and bind to E2 subunit, E2, E1, E2, E3, right? E2 subunit has lipoic acid, lipoate, and it's going to go and bind to that lipoate and inhibit the enzyme. Similarly, alpha ketoglutarate dehydrogenase enzyme complex is also inhibited by arsenic by binding to E2 subunit lipoic acid. It has SH group or thiol group. It will go and bind there. So that's how alpha ketoglutarate is conversion into succinyl-CoA is inhibited in the presence of arsenate. And also note that alpha ketoglutarate dehydrogenase complex, it needs E1, E2, E3 subunit, it needs 5 coenzymes. All that points you have to bring it here. Now second carbon is released here. So 2 carbons, acetyl-CoA, both the carbons are now released as carbon dioxide. That means acetyl-CoA is completely oxidized by the end of this step. Okay, 2 carbons are removed because acetyl-CoA has got only 2 carbons. So TCA cycle is a complete oxidation of acetyl-CoA. So rest of the carbons, that succinyl-CoA has got these 4 carbons, and these 4 carbons are actually from oxaloacetate carbons. Remember that. So what happens to the succinyl-CoA? Succinyl-CoA is converted to succinate done by succinate thiokinase enzyme. And now that succinate thiokinase, what it does, it's going to phosphorylate GDP and make it as GTP. This is an example for substrate level phosphorylation. So already we have seen 2 substrate level phosphorylation. One is at phosphoglycerate kinase step. Second is at pyruvate kinase step in glycolysis. Those 2 are the example for substrate level phosphorylation. Whereas here, conversion of succinyl-CoA to succinate, there is a formation of GTP done by succinate thiokinase. That's another example for substrate level phosphorylation without going into electron transport chain. Now, succinate is converted to fumarate by succinate dehydrogenase enzyme, and during this step there is a generation of FADH2 and succinate dehydrogenase. Special point about this enzyme is it is in fact a part of electron transport chain. In fact, it is one of the complex 2 in the electron transport chain. It is embedded in the inner side of inner mitochondrial membrane, whereas all other TCA cycle enzymes are there in the site of in the matrix of mitochondria. Remember that point. So FADH2 can go into electron transport chain and it is worth 1.5 ATP. And note that FADH2 means FAD gets into the reaction, FADH2 is made. So FAD is coming from riboflavin, so riboflavin deficiency can lead to decreased activity of succinate dehydrogenase enzyme in the TCA cycle. Now, fumarate is converted to malate by fumarase enzyme, not really that important, and malate is converted into oxaloacetate, and that is done by malate dehydrogenase enzyme, and there also you get one NADH plus H plus molecule. So overall, by the end of TCA cycle, oxaloacetate is regenerated because oxaloacetate entered in. At the end of the reaction, you are regenerating oxaloacetate. So what is consumed in the TCA cycle is only acetyl-CoA, which is oxidized into 2 carbon dioxide. So that means glucose making two pyruvates. These 2 pyruvates going into 2 acetyl-CoA, there will be release of 2 carbon dioxide. So 6 carbon now become 2 carbon. 2 acetyl-CoA molecule, 2 plus 2, 4 carbons are there. Now acetyl-CoA is going into one TCA cycle releasing 2 carbon dioxide, another TCA cycle releasing 2 more carbon dioxide. So 4 carbon dioxide released in the TCA cycle, 2 carbon dioxide released from pyruvate to acetyl-CoA, 2 pyruvate to 2 acetyl-CoA, total 6 carbon dioxide. Those are coming from 6 carbons from glucose. So that's a complete oxidation of glucose. Glucose to 2 pyruvate, 2 pyruvate to 2 acetyl-CoA, 2 TCA cycle, 6 carbon dioxide, complete oxidation. How much energy we are going to get? So already we have calculated 7 ATPs from glucose to 2 pyruvate under aerobic condition, and 2 pyruvate into 2 acetyl-CoA, we have 2 NADH plus, that means 5 ATP here, and 2 acetyl-CoA into 2 TCA cycle. Each TCA cycle we are going to get 3 NADH plus H plus molecule, 1 FADH2 and 1 GTP, and NADH plus is 2.5 ATP. So 2.5 plus 2.5 plus 2.5, 3 NADH, right? 2.5, 2.5, 2.5. So total 7.5, 1 FADH is 1.5 ATP. So, and then 1 GTP is equal to 1 ATP. So, total we are going to get 10 ATP per TCA cycle. So, how many TCA cycle? 2 acetyl-CoA means 2 TCA cycle. 2 TCA cycle means 20 ATP. 10 plus 10, 20. So, 20 here plus 5, this is 5. Pyruvate to acetyl-CoA 5 ATPs. 20 plus 5, 25 plus 7. So, how 7? 5 plus 2, 7. So, that is 32 ATPs. 20 plus 5, 25, 25 plus 5, 30, 30 plus 2, 32 net ATPs from 1 glucose into 6 carbon dioxide if a cell uses malate aspartate shuttle mechanism. Why? Because NADH plus going into electron transport chain using malate aspartate shuttle, you are going to get 2.5 ATP at this stage. If it uses glycerol 3-phosphate shuttle mechanism, you are going to get only 1.5 ATP. Detailed on that, I have a video, you can watch that video. So, shuttle mechanism video. Now, this is all about a TCA cycle. Now, that's we have converted totally glucose into pyruvate, pyruvate into acetyl-CoA, acetyl-CoA completely oxidized in the TCA cycle. That's complete oxidation of glucose. And then we have seen glucose to 2-lactate under anaerobic condition. So, that's about glycolysis, including link reaction and the TCA. So, now we have seen oxidation of glucose into pyruvate, pyruvate into acetyl-CoA, acetyl-CoA into TCA cycle. So, what we will do now is we will reverse some of these reaction and see how glucose can be synthesized. So, what I am trying to explain now to you is it is the gluconeogenesis. So, what is gluconeogenesis? Gluconeogenesis, it is the synthesis of a glucose molecule from non-carbohydrate sources. So, only few tissues in our body can conduct gluconeogenesis. The most important one is liver. To a certain extent, gluconeogenesis can go on in the kidney and also it can go on in the intestine. But the gluconeogenesis that is going on in the liver is the glucose that is made there is for other tissues. So, not for its own purpose. Okay. So, how glucose is made in gluconeogenesis? So, there are certain precursors for gluconeogenesis. What are the precursors for gluconeogenesis? The most important precursor for gluconeogenesis is the alanine. See, the alanine that is coming from protein degradation whenever proteins are undergoing turnover. So, alanine and other glucogenic amino acids, out of 20 standard amino acids, 18 of them can make glucose. Only 2 amino acids cannot make glucose, and that is leucine and lysine, leucine-lyse. That's a mnemonic you can remember, leucine and lysine. So, other than the leucine and lysine, all other amino acids can make glucose. So, one of the example that we are taking here is the alanine, because alanine is the predominant amino acid that comes out of the skeletal muscle degradation during fasting and starvation. So, we are taking alanine here. So, alanine by alanine transaminase reaction is converted to pyruvate. So, pyruvate formation is transamination from alanine to pyruvate. It is a transamination process which I am going to explain to you later. Now, pyruvate, once you get pyruvate, pyruvate can be reversed all the way to glucose, and that is one of the substrate for gluconeogenesis. Second substrate is lactate. Lactate can be reversed, converted back into pyruvate, and pyruvate can go back all the way into glucose. So, let us see how that happens. And first I will explain alanine and lactate, and then I will come to other two substrate for gluconeogenesis, that is propionyl-CoA, and the fourth one is glycerol. I will explain those two later. Now, let us finish lactate and alanine. So, in order to take lactate and alanine all the way to glucose, lactate is a 3-carbon molecule, and alanine is also, it will provide 3 carbon if you remove amino group. So, these two have 3 carbons, but glucose is 6 carbon. That means effectively we need 2 alanine or 2 lactate or 2 alanine, or 1 lactate 1 alanine making 2 pyruvates. All we need is 2 pyruvate. So, once we get those 2 pyruvates, so pyruvate can be converted back into phosphoenol pyruvate, and that does not happen directly like that, because pyruvate kinase is an irreversible step. So, remember, in order to understand gluconeogenesis, you really need to remember 3 irreversible reactions in glycolysis. If you can take care of those 3 irreversible reactions, rest all will be same reaction. Okay, what is that? So, the first irreversible reaction is glucokinase hexokinase reaction. Second is phosphofructokinase number 1, and the third is pyruvate kinase reaction. So these three irreversible reactions can be reversed by using a separate enzyme than the enzyme that is participating in glycolysis. So if you know those separate enzymes for these three irreversible reaction, you know gluconeogenesis. Let me take you into that aspect. So whenever a pyruvate is made, so this pyruvate has to be converted back into phosphoenol pyruvate. That is not possible by using pyruvate kinase because it's irreversible. And also note that gluconeogenesis is going on under fasting condition, where glucagon is more, insulin is less, and when there is more glucagon keeps pyruvate kinase inactive. So it is not going to convert phosphoenol pyruvate into pyruvate. Anyway, so what happens to the pyruvate? This pyruvate in the mitochondria, it is the mitochondrial matrix. It is going to be converted into oxaloacetate. Pyruvate is converted to oxaloacetate, and that will be done by enzyme pyruvate carboxylase. There is a carboxylation process. Carbon dioxide is added into this reaction, and that needs biotin as the coenzyme. So the pyruvate carboxylase enzyme is a gluconeogenic enzyme. It is induced by insulin. It is induced by glucagon. Insulin actually keeps this pyruvate carboxylase kind of low. So glucagon keeps this active. So thereby pyruvate is converted to oxaloacetate. So what happens to the oxaloacetate? Oxaloacetate is converted to malate. Oxaloacetate is converted to malate, and malate comes out of the mitochondria into the cytoplasm. So it will in the cytoplasm, it will be converted back into oxaloacetate. Okay, so all this is going on in the matrix of mitochondria. See, pyruvate which is made in the cytoplasm has to go down into the matrix of mitochondria because it cannot go back into phosphoenol pyruvate to reach glucose. That's not possible. So what happens? Pyruvate has to go down into the matrix of mitochondria, converted into oxaloacetate by pyruvate carboxylase enzyme. Oxaloacetate is converted to malate, and malate is coming out of mitochondria into the cytoplasm, because we have malate aspartate shuttle mechanism. Once malate is there in the cytoplasm, it is converted back into oxaloacetate in the cytoplasm, and this oxaloacetate is converted into phosphoenol pyruvate done by phosphoenol pyruvate carboxykinase enzyme. And during this reaction, there will be release of carbon dioxide, and it needs GTP, guanosine triphosphate, as the requirement. So this is what happens in the cytoplasm. So I am going to repeat one more time. Alanine in the cytoplasm converted to pyruvate by alanine transaminase enzyme. Lactate in the cytoplasm converted to pyruvate by lactate dehydrogenase enzyme. So what happens to the pyruvate? Pyruvate cannot go back into phosphoenol pyruvate to go all the way into glucose, because pyruvate kinase is not a reversible reaction. So how to make phosphoenol pyruvate? What is the way? So the way to do that is, pyruvate enters into the matrix of mitochondria, converted into oxaloacetate by pyruvate carboxylase enzyme using carbon dioxide, biotin, ATP and all, magnesium and all that. Once oxaloacetate made in the matrix of mitochondria, it is converted to malate by oxidizing NADH into NAD+, and malate is coming out of mitochondria into the cytoplasm, converted back into oxaloacetate in the cytoplasm, and oxaloacetate in the cytoplasm is now converted into phosphoenol pyruvate done by phosphoenol pyruvate carboxykinase enzyme, and that will release carbon dioxide, and it needs GTP to do that, because 5-carbon oxaloacetate is now 4-carbon oxaloacetate is now becoming 3-carbon phosphoenol pyruvate. So like this, 2 pyruvates will be converted into 2-phosphoenol pyruvate by indirect mechanism. So how many enzymes we have used to reverse pyruvate into phosphoenol pyruvate? Indirectly, we have used pyruvate carboxylase and phosphoenol pyruvate carboxykinase. These two are gluconeogenic enzyme number 1 and number 2, gluconeogenic enzymes to reverse pyruvate kinase. So reversal of pyruvate kinase needs 2 enzymes. One is pyruvate carboxylase, other is phosphoenol pyruvate carboxykinase. Okay. Now once we reverse that, we got phosphoenol pyruvate in the cytoplasm, 2 molecules, because 2 pyruvates are converted into 2-phosphoenol pyruvate, and now phosphoenol pyruvate is converted to 2-phosphoglycerate by enolase enzyme, reversible reaction. 2-phosphoglycerate to 3-phosphoglycerate, the mutase enzyme, and 3-phosphoglycerate to 1,3-bisphosphoglycerate by phosphoglycerate kinase enzyme. Of course, ATPs are used in the reaction, ADPs are released. I will come to that point later. Now 1,3-bisphosphoglycerate is converted to glyceraldehyde 3-phosphate 2 molecules done by glyceraldehyde 3-phosphate dehydrogenase reaction, which is reversible. And now once you get 2 molecules of glyceraldehyde 3-phosphate, in that one of the molecule is converted into dihydroxyacetone phosphate. Now we have one dihydroxyacetone phosphate, one glyceraldehyde 3-phosphate, they both combine. So 3 plus 3 carbons. So 6 carbons they combine to make 6 carbon fructose 1,6-bisphosphate. This will be done by aldolase enzyme, which is a reversible enzyme. So there is no need of special enzymes here. So all the same enzymes we are basically reversing. It's a concentration gradient effect. So basically these reversible enzymes, they work on concentration gradient. Now the gradient is to go from pyruvate all the way to glucose. That's why they are working in a reverse order. Now fructose 1,6-bisphosphate cannot be directly converted into fructose 6-phosphate. Why? Because in the glycolysis, that is a second irreversible reaction done by phosphofructokinase 1. So how to reverse that? So this PFK1 reaction here is irreversible. So we cannot use same PFK1 to reverse this reaction. So we need a separate enzyme to do that. So and that separate enzyme is fructose 1,6-bisphosphatase. Fructose 1,6-bisphosphatase enzyme, and this fructose 1,6-bisphosphatase enzyme, it is induced by glucagon, it is induced by cortisol, it is induced by epinephrine, all these gluconeogenic hormones. So they will induce fructose 1,6-bisphosphatase, take fructose 1,6-bisphosphate into fructose 6-phosphate. Of course, there will be release of one inorganic phosphate into the cytoplasm. That's how the irreversible, second irreversible reaction in glycolysis is reversed by using fructose 1,6-bisphosphatase. Now, fructose 6-phosphate can be converted into glucose 6-phosphate by phosphoglucoisomerase enzyme. It's a reversible reaction, and then glucose 6-phosphate cannot go into glucose by using glucokinase exokinase reaction. So that's the glucokinase exokinase reaction is irreversible reaction in glycolysis. So we need a special enzyme, and that special enzyme which will take glucose 6-phosphate into glucose, it is glucose 6-phosphatase enzyme. And this glucose 6-phosphatase enzyme, it is there in the lumen of endoplasmic reticulum. It is embedded into the towards the luminal side of endoplasmic reticulum. That means in the liver, kidney and intestine, glucose 6-phosphate has to go into the lumen of endoplasmic reticulum by T1 translocase transporter, and in the lumen of endoplasmic reticulum, where glucose 6-phosphatase enzyme is available attached to the endoplasmic reticular membrane, so it is going to release inorganic phosphate comes out and glucose is made, and this inorganic phosphate will come out into the cytoplasm by T2 translocase, and then glucose comes out into the cytoplasm from endoplasmic reticulum by T3 translocase. So this is what is the function of glucose 6-phosphatase enzyme. So deficiency of glucose 6-phosphatase because of mutation in G6PC gene, it can give rise to von Gerke disease, that is glycogen storage disease type 1A. If there is a mutation in a gene coding for T1 translocase, that is allowing glucose 6-phosphate to enter into the lumen of endoplasmic reticulum, that will give rise to glycogen storage disease 1B. So the difference between 1A and 1B is in 1B has all the signs of 1A. Additionally, there will be neutrophil dysfunction, neutropenia, bleeding disorders, infection risk, and all that will be there. That's about little bit clinical application. So that's the glucose 6-phosphatase there. Okay, so this is how alanine and lactate converted all the way into glucose by gluconeogenesis. So there are some special enzymes that I have mentioned in taking lactate and alanine all the way into glucose. Other than lactate dehydrogenase and alanine transaminase here, what are all the special enzymes that will take care of irreversible reactions in glycolysis? So those are, for pyruvate kinase irreversible step, we have pyruvate carboxylase, phosphoenol pyruvate carboxykinase, and for PFK1 reaction we have fructose 1,6-bisphosphatase, and for glucokinase exokinase step we have glucose 6-phosphatase. Remember these 4 enzymes, and all these 4 enzymes are induced by glucagon, induced by cortisol, induced by epinephrine. These are all glucogenic hormones. They will try to increase blood glucose level. Okay, so that's how alanine and lactate are converted into glucose. Let's little bit briefly, let me tell you how many ATPs are necessary to make one glucose from 2 lactate or 2 alanine. So let's get into that. So 2 lactate and 2 alanine going into 2 pyruvate, there is no ATP need there. Now 2 pyruvates going into 2 oxaloacetate, there is a need of ATP here. So 2 ATPs are necessary because 2 pyruvates to going into 2 oxaloacetate, 2 ATPs there, and then 2 oxaloacetate into 2 phosphoenol pyruvate, 2 GTPs. So 2 plus 2 there, 2 GTP means 2 ATP, 2 ATP, 2 GTP, so 2 plus 2, 4, plus now 2 phosphoenol pyruvate, 2 phosphoglycerate, 3 phosphoglycerate, and 1 3-bisphosphoglycerate. So there is a need of ATP here. 2 molecules of 3 phosphoglycerate, 2 molecules of 1 3-bisphosphoglycerate means 2 ATPs are needed there, that is plus 2 ATP. So up to this point we need ATP. So that means 6 ATPs are necessary to convert 2 lactate into 1 glucose or 2 alanine into 1 glucose. We need 6 ATPs. How do you get those 6 ATPs for gluconeogenesis? These 6 ATPs, they will come from beta oxidation of fatty acid. Remember that. So oxidation of fatty acid is the source for ATPs in gluconeogenesis in the liver. That's why whenever gluconeogenesis is going on, fatty acid oxidation is going on hand in hand to provide ATP. So gluconeogenesis breaks process. When gluconeogenesis process going on, adipose tissue lipolysis is going on because person is in fasting condition, starvation and all that, and in normal in a normal person. Okay, so this is the gluconeogenesis from lactate and alanine. Let me quickly take you into gluconeogenesis coming from propionyl-CoA all the way to glucose and glycerol all the way to glucose. Let's talk about propionyl-CoA. Here is the propionyl-CoA. So propionyl-CoA is a 3-carbon molecule. So how do you get propionyl-CoA? There are variety of sources for propionyl-CoA. So propionyl-CoA can come from mnemonic I have written here, that is Bob needs vitamin M, B-O-B, Bob needs vitamin M. So what is Bob needs vitamin M? So Bob needs vitamin M here, B for bile acid synthesis. I am going to explain you what is bile acid synthesis briefly. During bile acid synthesis, there is there will be release of propionyl-CoA. O for oxidation of odd number carbon fatty acid. Another B is oxidation of branched chain fatty acid, that is phytanic acid. And then VITM, that is valine, isoleucine, threonine and methionine. These are the amino acids. When they undergo oxidation or as a byproduct, they will be release of propionyl-CoA. So one of the product of oxidation of valine, isoleucine, threonine and methionine is propionyl-CoA. So propionyl-CoA come from Bob needs vitamin M. Remember that. Anyway, so once you get propionyl-CoA, propionyl-CoA is converted to methylmalonyl-CoA here, and that is done by propionyl-CoA carboxylase enzyme, and that needs ATP, biotin, carbon dioxide and magnesium. Now methylmalonyl-CoA converted into succinyl-CoA by methylmalonyl-CoA mutase enzyme, and that needs vitamin B12 in the form of deoxyadenosyl cobalamin. Once you get succinyl-CoA entering into TCA cycle, becoming succinate by succinate thiokinase, succinate to fumarate, fumarate to malate. Now you got malate. Malate moves out of mitochondria into the cytoplasm, becoming oxaloacetate, and oxaloacetate converted into phosphoenol pyruvate by phosphoenol pyruvate carboxykinase. It goes all the way to glyceroldiate 3-phosphate, dihydroxysteroid phosphate, fructose and 6-bisphosphate, and then fructose and 6-bisphosphatase take that into fructose 6-phosphate, glucose 6-phosphate, then glucose 6-phosphatase into glucose. So that's how propionyl-CoA goes all the way like this to glucose. So what are the important enzymes that you need to take propionyl-CoA all the way to glucose? It is propionyl-CoA carboxylase, methylmalonyl-CoA mutase, succinate thiokinase, TCA cycle enzyme, succinate dehydrogenase, then malate, oxaloacetate, so phosphoenol pyruvate carboxykinase, and then fructose and 6-bisphosphatase, glucose 6-phosphatase. These are some of the special enzymes, especially propionyl-CoA carboxylase, methylmalonyl-CoA mutase, succinate thiokinase, succinate dehydrogenase, and then phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, glucose 6 phosphatase. So what is not involved is this one. Pyruvate carboxylase is not involved because you are going to get malate to oxaloacetate. Oxaloacetate goes to phosphoenol pyruvate. So pyruvate carboxylase is not necessary for conversion of propionyl-CoA to glucose. So remember that. So that's how propionyl-CoA goes to glucose. Now come to glycerol. Glycerol we get from lipolysis of triacyl glycerol in the adipose tissue. Whenever person goes into fasting and starvation, adipose tissue undergoes lipolysis, breakdown of triacyl glycerol, and release 3 fatty acids and a glycerol molecule. And this glycerol reaching liver, glycerol undergoes glycerol kinase reaction, GK, glycerol kinase reaction, where ATP gets in, ADPs are released, all that. You get glycerol 3-phosphate, and that glycerol 3-phosphate is diverted into dihydroxyacetone phosphate by glycerol 3-phosphate dehydrogenase enzyme. Okay, so and then once you get dihydroxyacetone phosphate, so of course glycerol is a 3-carbon molecule, so you need 2 glycerols to make 1 glucose. So 2 glycerol to 2 glycerol 3-phosphate, 2 dihydroxyacetone phosphate, 2 in one of the dihydroxyacetone phosphate converted to glycerol dehyde 3-phosphate. They both combine to make fructose 1,6 bisphosphate, then fructose 6-phosphate, glucose 6-phosphate, and glucose. So like that, glycerol, conversion of 2 glycerol into 1 glucose, you need glycerol kinase, glycerol 3-phosphate dehydrogenase, fructose 1,6 bisphosphatase, and glucose 6-phosphatase. These are the 4 enzymes that are necessary here, important ones, to convert glycerol into glucose. So this is exactly how gluconeogenesis is going on, that is converting alanine, lactate, propionyl-CoA, glycerol all the way to glucose under fasting, starvation conditions and any catabolic conditions. Now, what is the regulation of gluconeogenesis? The important regulation of gluconeogenesis will go on in fructose 1,6 bisphosphatase step and pyruvate carboxylase step. How? So, fructose 1,6 bisphosphatase enzyme, the activity of this enzyme is regulated by one molecule, and that is fructose 2,6 bisphosphate. Fructose 2,6 bisphosphate. This fructose 2,6 bisphosphate will have a positive effect on PFK1 and it has got a negative effect on fructose 1,6 bisphosphatase. So, whenever there is high levels of fructose 2,6 bisphosphate, when do you see high levels of fructose 2,6 bisphosphate? Whenever there is more insulin, insulin is going to keep fructose 2,6 bisphosphate high. At that time, that high fructose 2,6 bisphosphate will keep PFK1 most active, thereby glycolysis is going on faster, glucose breaks down. That's what insulin does. So, insulin, by keeping higher levels of fructose 2,6 bisphosphate, it will keep PFK1 active, thereby glycolysis goes on. So, when glycolysis is going on, antagonistic pathway, that is gluconeogenesis, should not go on, right? So, at that time, so what this same fructose 2,6 bisphosphate, whenever it is at high concentration, it will have a negative effect on fructose 1,6 bisphosphatase enzyme. So, that's why gluconeogenesis goes down. So, glycolysis goes up, gluconeogenesis goes down, simultaneously regulated by single molecule, that is fructose 2,6 bisphosphate molecule. Okay, so it is going to act on 2 enzymes simultaneously. It has a positive effect on PFK1, negative effect on fructose 1,6 bisphosphatase. So, what happens under fasting, starvation, catabolic condition when glucagon, epinephrine? At that time, fructose 2,6 bisphosphate concentration decreases. So, when the fructose 2,6 bisphosphate concentration decreases, its negative effect on fructose 1,6 bisphosphatase is not there. So, this enzyme becomes free because fructose 2,6 bisphosphate is not there, right? So, enzyme becomes free. Its negative effect is not there now. So, enzyme is free to work. So, fructose 1,6 bisphosphate is free to work now. It will convert fructose 1,6 bisphosphate back into fructose 6-phosphate and reverse this glycolytic step back into glucose. So, whenever there is less amount of fructose 2,6 bisphosphate, its positive effect on PFK1 is also not there. So, glycolysis decreases. So, decreasing fructose 2,6 bisphosphate concentration, you are increasing the gluconeogenesis and decreasing the glycolysis process. That's how gluconeogenesis and glycolysis regulated simultaneously by a single molecule. And what is that? It is fructose 2,6 bisphosphate. Now, pyruvate carboxylase reaction can also be regulated, and pyruvate carboxylase enzyme is positively modulated by acetyl-CoA. Acetyl-CoA is an obligate activator of pyruvate carboxylase. Whenever person is in fasting condition and starvation, there will be elevated levels of acetyl-CoA, because I told you when gluconeogenesis is going on, beta oxidation is going on, and the product of beta oxidation is acetyl-CoA. So, there will be higher levels of acetyl-CoA, and that acetyl-CoA will keep pyruvate carboxylase most active. It is an obligate activator. Thereby, pyruvate is converted to oxaloacetate, and oxaloacetate can go back into gluconeogenesis process. So, that is another way of regulation of gluconeogenesis. So, that is about gluconeogenesis regulation and all the important substrate for gluconeogenesis. Now, let me take you into glycogen synthesis, glycogen metabolism, glycogen synthesis and glycogen degradation. Here is the glucose conversion into glucose 6-phosphate, and whenever there is higher levels of glucose 6-phosphate, when glucose 6-phosphate is kind of building up, if the glycolysis is saturated, when PFK1 is saturated, fructose 6-phosphate which accumulates will go into glucose 6-phosphate, and this glucose 6-phosphate cannot go into glucose, it is a irreversible step. So, what happens? Whenever glucose 6-phosphate kinds of building up, so this glucose 6-phosphate which is trapped here, because conversion of glucose to glucose 6-phosphate in the very step is a trapping of glucose inside the cell, it is committed to the cell now. So, glucose 6-phosphate is diverted into glucose 1-phosphate formation. So, now this glucose that is done by phosphoglucomutase enzyme. Glucose 1-phosphate, this glucose 1-phosphate is converted into UDP glucose by UDP glucose pyrophosphorylase enzyme. So, where UTP is converted to UDP and all that. So, glucose 1-phosphate converted into UDP glucose, and there is a lot of energy released in that reaction, because UTP breaks into UDP, and that UDP is released here, and inorganic phosphate and this phosphate. So, basically UTP getting into this reaction and releasing PPI, inorganic phosphate, which breaks into 2 inorganic phosphate molecules. It is a totally exergonic reaction, lot of energy released. So, you get UDP glucose there, and this UDP glucose will go and bind to glycogenin, and that is a protein. Glycogenin is a protein. So, UDP glucose goes and interact with glycogenin, especially the tyrosine, hydroxyl containing amino acid, aromatic hydroxyl amino acid, tyrosine. So, tyrosine is going to accept that glucose from UDP glucose. So, because every time UDP glucose interacts here, UDP is released, glucose will bind. So, glycogenin continuously accept that UDP glucose molecule, where UDP released, glucose binds and making a glycogen primer. Usually around 8 glucose residues are added here to make a glycogen primer, and this type of addition of glucose, UDP glucose to glycogenin protein is called as autoglycosylation process. Remember this. Autoglycosylation means there is no enzyme that is directly involved in this glycosylation process. Glycogenin is a protein, it itself acts as an enzyme to add UDP glucose molecules over its tyrosine residue. It acts as an enzyme itself. But only thing, the difference between glycogenin and a classic enzyme is, classic enzyme it comes out of the reaction unaltered, whereas glycogenin it is adding glucose over its own surface. So, it is not really a classic enzyme example, that is why it is just we refer it as autoglycosylation. So, once we get glycogen primer, this glycogen primer takes more UDP glucose and become a glycogen, that will be done by enzyme glycogen synthase. A glycogen synthase enzyme is going to take UDP glucose, release UDP and add only glucose molecule. Every time UDP will be released, UDP is not added. So, that is how glycogen is synthesized, and note that this glycogen synthesis, it needs another enzyme, because what glycogen synthase does is, it just makes alpha 1,4 glycosidic bond and make a linear chain, long linear chain is made by glycogen synthase enzyme. So, once the chain is elongated for around 11 to 14 glucose residue, some part of that glycogen chain is clipped by breaking alpha 1,4 bond and transferred to some other point by creating alpha 1,6 bond, And so that means breaking alpha 1 4 and creating alpha 1 6 bond, it will be done by enzyme called as branching enzyme. Branching enzyme, what it does? It is going to break alpha 1 4 bond and create alpha 1 6 bond. That's why it is referred as alpha 1 4 to alpha 1 6 transferase enzyme. Sometimes it is simply written as 4 6 transferase. So with the help of two enzymes, glycogen synthase and 4 6 transferase enzyme or branching enzyme, we are going to make highly branched glycogen structure. How many branch points? For every 9 to 10 glucose residues, there will be one branch point in a glycogen. So it's a highly branched glycogen molecule we are going to get, and there are so many non-reducing ends in a glycogen except glycogenin, where it is accepting a glucose. That was the reducing end, and that is also attached with the tyrosine. So in fact, there is no reducing end in a glycogen molecule. It's all non-reducing, and multiple non-reducing ends will be produced, and that's highly branched glycogen. So what is the use of highly branched glycogen? So having higher branch points, it makes it easier for the cell to degrade glycogen, release glucose molecules in a shorter time, and also when person goes into fed condition, it is easy to remake that glycogen in a shorter period of time. So that's why we have a non-reducing ends there. So once we have this glycogen, which is a highly branched structure made sufficiently synthesized in the liver and skeletal muscle. Why I have written only liver and skeletal muscle? Because quantitatively the concentration of glycogen is much higher in the liver compared to skeletal muscle. But the skeletal muscle quantity is more, that's why the skeletal muscle in terms of total quantity of glycogen in our body is much higher than the glycogen that is available in the liver. So in concentration wise, gram to gram ratio, liver has got higher glycogen concentration, whereas skeletal muscle has got higher glycogen quantity in our body. Remember that. Now, this all happens whenever person is in fed condition, when insulin is more, because insulin is going to keep glycogen synthase active by maintaining it in dephosphorylated condition. Remember that. Okay. So what happens when the person goes into fed condition? When person goes into what happens when the person goes into fasting condition? So when the person goes into fasting condition or starvation, not really starvation, because in the under starvation there is no glycogen left. So under fasting condition, what happens? So glucagon is increased, whether glucagon, or when person is exercising, epinephrine is increased. So either glucagon under fasting condition or epinephrine during exercise stressful situation, what these hormones will do? So glucagon binding to glucagon receptor, epinephrine binding to epinephrine receptor, if it is a beta receptor, beta adrenergic receptor for epinephrine, or glucagon G protein coupled receptor. So both of them, what they do, they are going to activate protein kinase A enzyme by increasing adenylate cyclase, cyclic AMP, and then protein kinase A. And this protein kinase A, what it does, it's going to phosphorylate this glycogen synthase enzyme and keep it inactive, and going to phosphorylate glycogen phosphorylase enzyme. And this glycogen phosphorylase enzyme is phosphorylated by protein kinase A enzyme. So, and the phosphorylated form of glycogen phosphorylase is an active enzyme, and what it does, it's going to add inorganic phosphate into this reaction and release glucose 1-phosphate. That's what I have written here. So it's going to release glucose 1-phosphate. So glycogen breaks into glucose 1-phosphate after the addition of inorganic phosphate by glycogen phosphorylase enzyme. So inorganic phosphate, it acts as a nucleophile and break that alpha 1,4 bond and release glucose as glucose 1-phosphate, not as directly glucose. And this glycogen phosphorylase will do until 4 glucose residues are left at the branch point. Because when the branch has 4 glucose on either side, there will be steric endurance for glycogen phosphorylase for one of the branch to enter into the active site of glycogen phosphorylase, because they both are equal, they cannot go into the active site one at a time. So at that time glycogen phosphorylase action will stop. Now comes the debranching enzyme. So we have a debranching enzyme. I will write as a debranching here. Debranching enzyme. So what is the function of debranching enzyme? Debranching enzyme has 2 enzymatic function. It has 4,4 transferase function, 4,4 transferase, and also it has got alpha 1,6 glucosidase function. So 4,4 transferase, what it does, out of 4 glucose residue in each branch point, So 4,4 transferase, what it does, it's going to clip that alpha 1,4 bond of the 3 glucose. So 1, 2, 3, 4, 1, 2, 3, 4. 3 glucose residue is taken out from 4 glucose and by breaking alpha 1,4 bond and transfer it to this immediate second one or some other branch point there. So thereby elongating the other one by 3 more glucose. So it breaks alpha 1,4 bond and creates alpha 1,4 bond. That's why it is called as 4,4 transferase. I have a detailed video on this if you are not understanding me. So please watch my glycogen metabolism video now for better understanding if you did not get it. And then once 3 glucose residue taken from one of the branch point, there is only one glucose that is left at the branch point with alpha 1,6 linkase, and that is clipped by alpha 1,6 glucosidase enzyme, part of debranching enzyme, and that's what releases glucose directly. So majority of glycogen breaks into glucose 1-phosphate, only minor glycogen breaks into glucose directly. So for every 7 to 9 glucose 1-phosphate molecules, you're going to get 1 glucose, remember that, in glycogen degradation, and that's because of the number of glucose present in each branches. Now what happens to this glucose 1-phosphate coming from glycogen degradation? Glucose 1-phosphate is converted to glucose 6-phosphate. Glucose 6-phosphate gets into the lumen of endoplasmic reticulum, converted into glucose by glucose 6-phosphatase, and that glucose enters into the blood. So this glucose can go into blood and maintain the blood glucose level, especially if it is coming from liver glycogen degradation. So deficiency of any of these, some of these enzymes in the glycogen degradation, it can lead to glycogen storage diseases. What all that? So if there is a deficiency of glucose 6-phosphatase, one of the glycogen degradation enzyme, indirectly, it leads to Von-Gerke disease. And if there is before that some of the glycogen in the cytoplasm, glycogen granules in the cytoplasm are diverted into lysosomes. Some of the glycogen is like around 3% of the glycogen in the cytoplasm normally undergoes cell turnover, making degradation in the lysosome, and the lysosomal glycogen degradation enzyme is called as acid maltase enzyme. And this acid maltase, what it does, it will break alpha 1,4 bond, it will break alpha 1,6 bond and release glucose directly, and that's how glycogen is degraded into glucose directly in the lysosomes, done by acid maltase enzyme. There are two acid maltase enzyme for glycogen degradation. One is alpha 1,4 acid alpha 1,4 glycosidase, acid alpha 1,6 glycosidase. Don't confuse alpha 1,6 glycosidase part of debranching enzyme. It's a cytoplasmic enzyme, whereas the lysosomal enzyme is an acid stable acid alpha 1,6 glucosidase. Okay, so if there is a deficiency of acid maltase enzyme, it will give rise to Pompe disease. Remember that. And then if there is a deficiency of branching enzyme in the glycogen synthesis, it leads to Anderson's disease. If there is a deficiency of debranching enzyme, it will give rise to Cori disease. If there is a deficiency of glycogen phosphorylase enzyme in the muscle, it will give rise to McArdle's disease. If there is a deficiency of glycogen phosphorylase in the liver, it will give rise to Hirsch's disease. If there is a deficiency of phosphofructokinase 1 enzyme in the muscle and red blood cell, it will give rise to Torres disease. So these are some of the glycogen storage disorders I have briefly explained. So I have a detailed video on glycogen storage disorders, so you can watch that video if you are not sure about all the disorders. Now that's about glycogen degradation there. Now what happens to this glucose 6-phosphate additionally? So I have told you glucose 6-phosphate going into glucose 1-phosphate UDP glucose and then glycogen formation I have covered. Before I go to other side, let me just finish this part here. Now the glucose 1-phosphate, which is making UDP glucose, and this UDP glucose not only goes into glycogen synthesis, it can also make UDP glucuronate. This UDP glucuronate, that's basically oxidation of UDP glucose into UDP glucuronate. UDP glucuronate is converted to glucuronate, and glucuronate converted to L-gulanate. L-gulanate converted to L-xylulose. L-xylulose is converted to L-xylitol. So conversion of L-xylulose into L-xylitol can be deficient in some people, and that will give rise to essential pentasuria. Anyway, L-gulanate in other than humans, it can be converted to L-ascorbate. So that's how vitamin C can be synthesized in primates, but humans cannot synthesize because we are lacking some enzyme there. Now, and then UDP glucose convert into UDP glucuronate. This UDP glucuronate, it can also participate in detoxification process. It also participate in bilirubin conjugation process, like conversion of unconjugated bilirubin into conjugated bilirubin is using UDP glucuronate. And also drug detoxification can use UDP glucuronate. That's other uses of UDP glucuronate. Now, UDP glucose can also participate in galactose metabolism. Let me touch upon that, the minor carbohydrate metabolic pathway. So, galactose coming from lactose in the milk. Lactose by lactase enzyme breaks into glucose and galactose. Galactose is taken up by the cells, especially the liver, and galactose in the liver and even other tissues also. Galactose is converted to galactose 1-phosphate by galactokinase enzyme by using ATP. So that means galactose is trapped, inorganic phosphate is attached to the first carbon, inorganic phosphate is trapped, and the galactose 1-phosphate undergoes galactose 1-phosphate uridyl transferase enzyme, GALT enzyme, galactose 1-phosphate uridyl transferase enzyme. That is this reaction, second reaction here, where galactose 1-phosphate, it is going to interact with UDP galactose. Galactose 1-phosphate, it is going to interact with UDP glucose to make UDP galactose and release glucose 1-phosphate. That's how the condensation will go on. So, galactose 1-phosphate is going to condense with UDP glucose, and UDP glucose is that UDP part here will be given to this galactose, that becomes UDP galactose, and the phosphate here, 1-phosphate, first carbon phosphate, is attached to glucose, and that makes glucose 1-phosphate. Okay. So, overall what happened here, galactose 1-phosphate is now in the form of UDP galactose, and this phosphate is there in the form of glucose 1-phosphate. Okay. Now, what happened to this UDP galactose? We are interested in galactose, not glucose. So, in the very first reaction, means in the second reaction, you need UDP glucose, of course, that will come from glucose 1-phosphate, UDP glucose and all that. Once you get that UDP glucose, so the UDP glucose pyrophosphorylase enzyme, galactose 1-phosphate uridyl transferase enzyme is going to convert galactose 1-phosphate into UDP galactose, and this UDP galactose undergoes epimerase reaction, 4-epimerase reaction, and that is this third reaction here. So, this is second and first. First is galactokinase, second is galactose 1-phosphate uridyl transferase, third is 4-epimerase. So, what 4-epimerase does? 4-epimerase converts UDP galactose into UDP glucose, and now all the carbons of this glucose is actually from galactose. So, basically galactose 1-phosphate, UDP galactose and UDP glucose. And this UDP glucose in the second galactose metabolism, it will go into galactose 1-phosphate uridyl transferase reaction and convert it to UDP galactose and glucose 1-phosphate. And that glucose carbons in the second galactose metabolism will be coming from the first galactose carbons. And glucose 1-phosphate now, it can be converted into glucose 6-phosphate, and that glucose 6-phosphate can go into glycolysis if it is a skeletal muscle and other tissues, or it can be converted back into glucose, and that's how galactose can be converted to glucose indirectly. You cannot convert galactose directly into glucose, that's not possible. So, that's why galactose under fasting condition, if the milk is taken, galactose can be converted to glucose by going through all this mechanism. So, what are the important things here? Deficiency of galactokinase enzyme, it will give rise to a disease called galactokinase deficiency, sometimes referred as non-essential galactosemia. Most importantly, the deficiency of galactose 1-phosphate uridyl transferase, it will give rise to classic galactosemia, where mental retardation is there, bilateral cataracts, hepatomegaly, jaundice and all those signs will be seen. So, that's about galactose metabolism briefly. Now, let me take you into pentose phosphate pathway. Now, the pentose phosphate pathway, it is a conversion of glucose 6-phosphate all the way into 6-phosphogluconate. That will be done by oxidative phase of pentose phosphate pathway. Oxidative. So, pentose phosphate pathway, it is broken down into oxidative phase and non-oxidative phase. In oxidative phase what happens? So in oxidative phase what happens? Glucose 6-phosphate is converted to 6-phosphogluconate. That is done by two enzymes. Very first enzyme is glucose 6-phosphate dehydrogenase enzyme, and the second enzyme is 6-phosphogluconate dehydrogenase enzyme. So glucose 6-phosphate dehydrogenase, 6-phosphogluconate dehydrogenase, so both of them what they will do? They will release NADPH plus H plus molecule, and also there will be release of carbon dioxide. Okay, so okay, not here. So the first reaction done by glucose 6-phosphate dehydrogenase, here NADH plus released and 6-phosphogluconate released, and now the 6-phosphogluconate dehydrogenase will release one more NADPH plus and carbon dioxide. So that means 6 carbon glucose 6-phosphate, which is a hexose monophosphate. You know, glucose is hexose and one phosphate is attached to the 6th carbon. That's what is hexose monophosphate, and this hexose monophosphate is shunted, instead of glycolysis it is shunted towards this side. That is why this pathway is called as HMP shunt. HMP shunt means hexose monophosphate is shunted towards this side rather than glycolysis, because there is accumulation of glucose 6-phosphate there, simply because glycolysis is saturated. Now 6-phosphogluconolactane to ribulose 5-phosphate, which is a 5-carbon sugar. Now the 5-carbon ribulose 5-phosphate coming from 6-carbon glucose 6-phosphate, and this 5-carbon ribulose 5-phosphate is converted into 5-carbon ribose 5-phosphate. 5-phosphate can go into nucleotide biosynthesis. Why? Because this can make PRPP, phosphoribosyl pyrophosphate. Ribose 5-phosphate can be converted into phosphoribosyl pyrophosphate, and note that phosphoribosyl pyrophosphate is a positive activator on the de novo synthesis of purines and also pyrimidine biosynthesis. Purine and pyrimidine biosynthesis. So PRPP is a positive modulator on purine biosynthesis, pyrimidine biosynthesis. So there will be elevation of both purines and pyrimidines. So that's how ribose 5-phosphate goes into nucleotide biosynthesis. Remember, whenever there is higher levels of pentose phosphate pathway, there will be more purines and pyrimidines, and that can lead to hyperuricemia. Remember that. Now, so the pentose phosphate pathway, this is also called as PPP, pentose phosphate pathway. Why pentose phosphate pathway? Because ribose 5-phosphate is a pentose and it is a phosphate is attached. So pentose phosphate pathway. Now, so what happens to the ribulose 5-phosphate when ribose 5-phosphate is made, and if the cell don't want nucleotide biosynthesis? Like if the cell only wants NADPH plus but not ribose 5-phosphate, like red blood cells, right? Red blood cells don't need purines and pyrimidines and all that. So it needs NADPH plus to maintain the membrane integrity and all that, because NADPH plus is necessary to maintain glutathione in reduced state, that is GSH maintenance. Otherwise, if the GSH levels are less, there will be lipid peroxidation, red blood cell breaks open, and that can lead to hemolytic anemia. That's what happens in glucose 6-phosphate dehydrogenase deficiency. Okay, so ribose 5-phosphate, ribulose 5-phosphate, if the cell don't want this nucleotide biosynthesis, so they can go back into fructose 6-phosphate formation or glycerol 3-phosphate formation, and that will be done by non-oxidative phase of pentose phosphate pathway. So what are all the important points that you should remember in non-oxidative phase of pentose phosphate pathway? In non-oxidative phase of pentose phosphate pathway, you got to remember some intermediates, names of the intermediates, and also you got to remember 2 enzyme names. In that, one enzyme name is most important. What are the intermediates? We have xylulose 5-phosphate, cedoheptulose 7-phosphate, right? So ribulose 5-phosphate, xylulose 5-phosphate, cedoheptulose 7-phosphate, and also erythrose 4-phosphate. Erythrose 4-phosphate. So these are the intermediates that you should remember, just in case. Okay, ribulose 5-phosphate, xylulose 5-phosphate, cedoheptulose 7-phosphate, erythrose 4-phosphates are the intermediates. So what are the enzymes in the non-oxidative phase of pentose phosphate pathway? Enzymes are transaldolase and transketolase. Transketolase enzyme is important because transketolase enzyme needs thiamine pyrophosphate as the coenzyme. It's a derivative of thiamine. So thiamine deficiency will lead to decreased activity of transketolase, so that will affect non-oxidative phase of pentose phosphate pathway. And also, what is the importance of non-oxidative phase of pentose phosphate pathway? When opposed to oxidative phase, it is non-oxidative phase of pentose phosphate pathway reactions, they are reversible reactions, as you see the arrow here, both side bidirectional, reversible. That means ribulose 5-phosphate can go all the way into fructose 6-phosphate and glycerol D8-3-phosphate after combination of 2-3 reactions, or fructose 6-phosphate and glycerol D8-3-phosphate, they can come into pentose phosphate pathway through non-oxidative phase. So that this reversible thing is there. Why this happens? Because if the cell wants ribose 5-phosphate, whenever there is excess NADPH plus, so note that higher levels of NADPH plus will have a negative effect on oxidative phase of pentose phosphate pathway, because the most regulated enzyme or the important regulatory enzyme in pentose phosphate pathway is glucose 6-phosphate dehydrogenase enzyme, and this glucose 6-phosphate dehydrogenase enzyme is negatively modulated by higher levels of NADPH plus, and this enzyme is induced by insulin, remember. So whenever person is in fed condition, insulin keeps higher levels of oxidative phase of pentose phosphate pathway, because NADPH plus is consumed for fatty acid synthesis, cholesterol biosynthesis and all that. Anyway, so and otherwise normally there will be higher levels of NADPH plus compared to NADP plus for 100:1. There will be, for every 100 molecule of NADPH plus there will be 1 molecule of NADP plus. So always glucose 6-phosphate dehydrogenase is kind of negatively modulated. But whenever there is a reductive biosynthesis is going on, like cholesterol biosynthesis, fatty acid synthesis, a nucleotide biosynthesis, or when there is especially deoxynucleotide biosynthesis, or when there is a bile acid synthesis. So whenever detoxification, whenever it is going on, so when bacterial killing mechanism is going on, like NADPH oxidase bacterial killing mechanism, or nitric oxide synthesis, all these are the process, and GSH maintenance. So all these are processes where NADPH plus are necessary. When those processes are going on, oxidative phase has to go on at a higher level, because NADPH plus will be consumed, glucose 6-phosphate dehydrogenase enzyme will be more active. So more and more oxidative phase will go on to regenerate that NADPH plus molecule. Okay, so what is the clinical application of this? So the clinical application of pentose phosphate pathway is the deficiency of glucose 6-phosphate dehydrogenase enzyme, and it's an X-linked recessive condition. So that means males are the ones where symptomatic and females are acting as a carriers. Usually it is seen in people living in Mediterranean region, then the Asians, Africans, where malaria is endemic, where glucose 6-phosphate dehydrogenase enzyme deficiency can be seen. So this is a deficiency. That means some amount of GSH is still there because of the residual enzyme activity that is available. So whenever person goes into like excess reactive oxygen species generation, like becoming infected on certain drugs like antimalarials, sulfonamides, analgesics, things like that, or naphthalines, or some chemicals where reactive oxygen or fava bean consumption. So during that time disease becomes triggered, like they become symptomatic. So becoming symptomatic upon consumption of certain things, that will, or becoming infected with a malaria or something like that, it makes a person symptomatic, and signs and symptoms of hemolytic anemia will occur. Why there will be hemolytic anemia in glucose 6-phosphate dehydrogenase deficiency? Because NADPH levels will be less. So less NADPH means less GSH, that is reduced glutathione. Less reduced glutathione means lipid peroxidation will go on unhindered, because GSH will prevent membrane lipid peroxidation. So membrane is undergoing peroxidation, membrane permeability increases, and membrane will allow things to get in, and especially in the red blood cell will swell, osmotic effect will happen, it will burst open and lead to hemolysis. So hemolytic anemia. And also more reactive oxygen species means hemoglobin is denatured and make a hinge body. So hinge bodies are one of the finding that you see in glucose 6-phosphate dehydrogenase deficiency. Hinge bodies are also referred as bite cells under when you observe under microscope. That's a little bit about clinical aspect. Transketolase enzyme deficiency of thiamine pyrophosphate or deficiency of thiamine can be indirectly measured by measuring red blood cell transketolase activity. That's about pentose phosphate pathway. So one of the small little connection that I would like to make here is the fructose metabolism. So the fructose metabolism here, I have written it here. So the fructose is converted into fructose 1-phosphate by fructokinase enzyme, and this fructose 1-phosphate here, fructose to fructose 1-phosphate by fructokinase enzyme, and the fructose 1-phosphate breaks into glyceraldehyde and dihydroxyacetone phosphate done by aldolase B enzyme. Especially this happens in the liver, and dihydroxyacetone phosphate can go into glycerol 3-phosphate and esterified to make triacylglycerol, and that's how it is diverted. Fructose is diverted into triacylglycerol formation, and also glyceraldehyde here can be converted into glyceraldehyde 3-phosphate, and that glyceraldehyde 3-phosphate can go into glycolysis to make pyruvate, pyruvate to acetyl-CoA, and acetyl-CoA becoming citrate. Okay, so whenever there is excess consumption of fructose, it will promote triacylglycerol formation, esterification, and also it will promote more citrate formation. And especially if the fructose is consumed after meals when thesis cycle is kind of saturated, this citrate, it can move out of mitochondria, as I have shown here. It can move out of mitochondria into the cytoplasm, and in the cytoplasm it will break into acetyl-CoA and oxaloacetate. So when acetyl-CoA is there in the cytoplasm, oxaloacetate is there in the cytoplasm. So this acetyl-CoA, I will go about acetyl-CoA only. Oxaloacetate I will talk about later. Oxaloacetate will become malate, malate to become pyruvate by malic enzyme. So malate to pyruvate by malic enzyme, and that also releases NADPH plus in the cytoplasm. So citrate breaks into acetyl-CoA and oxaloacetate whenever TCA cycle is saturated. When citrate builds up, citrate moving into the cytoplasm breaks open into acetyl-CoA and oxaloacetate by citrate lyase enzyme, and acetyl-CoA, it has two direction to go. One is acetyl-CoA can become malonyl-CoA by acetyl-CoA carboxylase. Now I am taking you into fatty acid synthesis. Okay, so acetyl-CoA into malonyl-CoA and the acetyl-CoA carboxylase enzyme, and that's the regulated enzyme in fatty acid synthesis. Insulin keeps acetyl-CoA carboxylase active by keeping it in dephosphorylated state. Glucagon keep it inactive by maintaining it in phosphorylated state. Even epinephrine will also do that. Now once you get conversion of acetyl-CoA into malonyl-CoA, acetyl-CoA is a 2-carbon molecule, malonyl-CoA is a 3-carbon molecule done by acetyl-CoA carboxylase. Now malonyl-CoA will go into fatty acid synthase complex. Okay, and in the fatty acid synthase complex, malonyl-CoA will contribute 2 carbons for the elongation of the fatty acid chain, and you get a fatty acid. Usually it is a 16-carbon palmitic acid. Okay, once you get 16-carbon palmitic acid, basically there are certain steps involved here in the fatty acid synthase complex, and 4 important ones that I would like to mention is condensation and decarboxylation, reduction, dehydration, and reduction. Okay, in condensation and decarboxylation, there will be release of carbon dioxide from malonyl-CoA. One of the carbon released, only 2 carbons are added. So malonyl-CoA combines with acetyl-CoA to make a 4-carbon molecule. So, and that 4-carbon molecule will undergo reduction and then dehydration and reduction. In that reduction step, there are 2 reduction steps for addition of every 2 carbons in a fatty acid growing chain. So in the reduction step, there is a need of NADPH plus, and that NADPH plus will come from, majority of it will come from pentose phosphate pathway, some of it will come from malic enzyme, conversion of malate to pyruvate, and also there is one more source of NADPH plus in the cytoplasm, that is cytoplasmic isocitrate dehydrogenase. Okay, so these are 3 sources of NADPH plus in the cytoplasm. One is pentose phosphate pathway oxidative phase. In that there are 2 enzymes, glucose 6-phosphate dehydrogenase, 6-phosphogluconolactone dehydrogenase, and then the malic enzyme, and the third is cytoplasmic isocitrate dehydrogenase enzyme. Anyway, so that's how fatty acid is synthesized, 16 carbon. If the cell wants more carbons, like 18 carbon, 20 carbon, so it can be elongated. Usually elongation goes on in the endoplasmic reticulum, which is called as microsomal elongation system. Malonyl-CoA is a 2-carbon donor there. There is a mitochondrial elongation system also, where acetyl-CoA is a 2-carbon donor. Remember that difference. Now, what happens to the fatty acid? Fatty acid, if it is made in the liver, usually predominantly it is made in the liver. So what liver will do with the fatty acid? Fatty acids are diverted into the lumen of endoplasmic reticulum, where glycerol 3-phosphate is available coming from dihydroxyacetone phosphate. That's why glycolysis, you know, glucose dihydroxysteroid phosphate. Some of that DHAP is diverted into glycerol 3 phosphate, and that glycerol 3 phosphate in the endoplasmic reticulum, it is esterified with the fatty acid to make triacyl glycerol. 3 fatty acids are attached to 3 carbons in glycerol 3 phosphate. First carbon, second carbon, third carbon phosphate removed and added with the fatty acid. So 3 fatty acids are added to glycerol backbone to make a triacyl glycerol. Very first step in the esterification process done by glycerol 3 phosphate acyl transferase enzyme, and that particular enzyme is induced by insulin and it is kept in dephosphorylated state by insulin molecule. Thereby insulin promotes triacyl glycerol synthesis. Note that insulin, it will promote fatty acid synthesis under well-fed condition when plenty of glucose is available by keeping all the enzymes starting from glucose, important ones, active in dephosphorylated state and also inducing those enzymes, especially in the liver. I am going to review that for you. Glucokinase is induced by insulin under well-fed condition in the liver. PFK1 kept active by insulin by keeping fructose 2,6 bisphosphate higher by keeping PFK2 kinase subunit dephosphorylated. And then it is going to keep pyruvate kinase active. Insulin is going to keep pyruvate kinase active, thereby more pyruvate is made. And then it is going to keep pyruvate dehydrogenase complex active by keeping it in dephosphorylated state, inducing that enzyme. And then it is going to express increased citrate transporters, and in the cytoplasm it is going to induce citrate lyase enzyme, and then it is going to induce acetyl-CoA carboxylase enzyme, most regulated enzyme in fatty acid synthesis, and also it maintains acetyl-CoA carboxylase in dephosphorylated state, that is an active form of an enzyme, and it is going to induce fatty acid synthase enzyme also. It is going to induce malic enzyme which is providing NADPH plus for fatty acid synthesis, and it is going to induce glucose 6-phosphate dehydrogenase enzyme which is a major provider of NADPH for fatty acid synthesis. And also note that for every 2 carbons that are condensed, 2 NADPH plus molecules are necessary. That means to synthesize 16 carbon palmitic acid, there will be necessary of around 14 NADPH plus 14 NADPH and 14 H plus molecules. And also acetyl-CoA to malonyl-CoA, there is a need for 1 ATP because it is done by acetyl-CoA carboxylase. Carboxylases, they need ATP, carbon dioxide and magnesium and biotin. So need of ATP there. So for making 16 carbon palmitic acid, there is a need of 7 malonyl-CoAs, because how do you find out how many NADPH, how many malonyl-CoAs are needed? If you know the number of carbons in a fatty acid, so here it is 16, divide that 16 by 2, so 16 divided by 2 is 8, so 8 minus 1, that is 7, so 7 malonyl-CoAs and 7 NADPH plus 14 NADPH plus 14 H plus molecules. Anyway, so that is about fatty acid synthesis there. Now let me take you into cholesterol biosynthesis. So acetyl-CoA converted into mevalonate. Acetyl-CoA condensed with another acetyl-CoA molecule to make acetyl-CoA. That condensed with one more acetyl-CoA molecule to make HMG-CoA, and HMG-CoA undergoes a reduction process to make mevalonate done by HMG-CoA reductase enzyme. And the most regulated enzyme in cholesterol biosynthesis is HMG-CoA reductase enzyme. And this HMG-CoA reductase enzyme is inhibited by statin group of drugs. Okay, so and also HMG-CoA reductase is inhibited by excess cholesterol, excess bile acids. Even the mevalonate, whenever it is accumulating, it will have a immediate feedback negative effect on HMG-CoA reductase enzyme. Now the mevalonate undergoing further series of reactions to make farnesyl pyrophosphate, and farnesyl pyrophosphate going into lanosterol. Lanosterol becoming cholesterol. Lanosterol is the first sterol, 30 carbon sterol molecule, where there will be release of 3 carbon dioxides making a 27 carbon cholesterol, and all the 27 carbons present in cholesterol molecule, they are all coming from acetyl-CoA molecule. Remember, so acetyl-CoA to mevalonate, mevalonate to farnesyl pyrophosphate, lanosterol and cholesterol. Okay, cholesterol in the liver can be esterified with the fatty acid in the endoplasmic reticulum to become cholesterol ester, and this cholesterol ester can be stored, can be loaded onto APO B100 to make VLDL molecule, and also excess cholesterol can be excreted in the bile as biliary cholesterol. So, and some of the cholesterol in the liver can go towards bile acid synthesis. Let me quickly review that. So, cholesterol undergoing 7-alpha hydroxylase reaction, regulated enzyme in bile acid synthesis, becoming 7-alpha hydroxycholesterol, and then it will break into choline-CoA and cyanodeoxycholine-CoA upon removal of 3-carbon propionyl-CoA. That's what is the Bob needs vitamin M, bile acid synthesis, propionyl-CoA release, 3-carbon. So, choline-CoA and cyanodeoxycholine-CoA, and these molecules, they will undergo conjugation process by glycine and taurine. Glycine and taurines are the conjugating molecules for bile acids. So, glycine conjugated product is glycocolate and glycosinodeoxycholate. Taurine conjugated molecules are taurocolate and taurosinodeoxycholate. So, purpose of this conjugation is to increase the emulsification process during lipid digestion and absorption process. So, that is what is one of the derivative of cholesterol here. It is this bile acids which are necessary for lipid digestion and absorption process. Okay, so there are some things that can come from farnesyl pyrophosphate other than cholesterol. Farnesyl pyrophosphate can give dolicol. Dolicol is participating in N-linked glycoprotein and proteoglycan synthesis, and also ubiquinone. Ubiquinone is one of the mobile complex in the electron transport chain, etc. So, ubiquinone and dolichols are the derivative of farnesyl pyrophosphate. That means whenever person is on HMG-CoA reductase inhibitors like statins, so there will be decrease in mevalonate, means decrease in farnesyl pyrophosphate, that means decrease in cholesterol. That's okay, that's what we wanted, but this decreases dolicol and also ubiquinone. HMG-CoA reductase and reductase drugs, HMG-CoA reductase inhibitors like statins, they will decrease not only cholesterol but ubiquinone, mobile complex in the electron transport chain, and also dolicol, thereby giving rise to some of the side effects of statins like rhabdomyolysis, muscle pain, myalgia also can be because of decrease in ubiquinone, where electron transport chain can be decreased. So, that is about fatty acid synthesis and cholesterol biosynthesis. Now, what happens to the triacylglycerol that is made in the endoplasmic reticulum, and also cholesterol ester that is made here, so and some of the phospholipids that are made. So, what happens to that? Alright, so now cholesterol ester and triacylglycerol that is made in the liver, and some of the phospholipid, vitamin E, vitamin K, all that, it will be loaded on to ApoB100 protein in the liver. You remember, like ApoB100 is coded by ApoB100 mRNA, means ApoB100 gene, ApoB100 mRNA in the liver, and same thing is undergoing mRNA editing in the intestine. I will come to that point later. So, ApoB100 accepting that cholesterol ester, triacylglycerol, phospholipid, vitamin E, vitamin K in the liver, making VLDL, very low density lipoprotein, and that is the nascent or a newly synthesized VLDL from the liver, which is secreted into the circulation, and this VLDL in the circulation, it will gain ApoC2 and ApoE from HDL in the circulation, and that is how nascent VLDL is matured in the circulation upon getting ApoC2, ApoE and other apolipoprotein from HDL molecule, remember. So, once VLDL is matured, so this VLDL in the circulation, it will undergo metabolism. So, VLDL is undergoing metabolism in the circulation, and that is initiated by enzyme lipoprotein lipase enzyme, that is LPL. This lipoprotein lipase enzyme, it is attached to the endothelial capillary, and that is with the help of heparan sulfate over the membrane. Remember that point. So, and the LPL, it is activated by ApoC2, which is present over VLDL molecule. So, what is the function of lipoprotein lipase? Lipoprotein lipase, as it is there in the name, it is going to act on the triacylglycerol present in VLDL. Remember, I am just explaining you lipoprotein metabolism. So, there are different type of lipoproteins, that is chylomicrons, VLDLs, IDL, LDL and HDL molecules. So, chylomicron is the biggest one, HDL is the smaller one, and chylomicron is super rich in triacylglycerol, around 90% of triacylglycerol in the chylomicron, and then 60% of triacylglycerol is there in the VLDL molecule. So relatively VLDL is rich in triacylglycerol, and then cholesterol ester is present in LDL. HDL has got phospholipid, cholesterol ester and all that. So according to the density, chylomicron is the least dense molecule, HDL is the most dense molecule. If you go from least dense to most dense, then chylomicron, VLDL, IDL, LDL, HDL. So, or if you go from most dense to least dense, you go in a reverse order. Okay. So, now that kind of VLDL which is made in the, remember, VLDL is synthesized and secreted by the liver, and it has ApoB100, ApoC2, ApoE as the major ones. Okay. And so, ApoC2 acting as an actuator of LPL, where triacylglycerol, most of it is degraded into free fatty acid and a glycerol molecule. And now what happens to the free fatty acid? Free fatty acids are usually taken up by the tissues which are beneath that endothelial capillaries. Every tissue has got their specific capillaries being expressed, and specific enzymes and different types of isoenzymes of lipoprotein lipases are expressed by the capillaries which are over the tissues which are beneath it. Okay, so example, lipoprotein lipase that is present over the endothelial capillary of adipose tissue is quite different than lipoprotein lipase that is present over the capillaries of cardiac tissue. So, cardiac tissue lipoprotein lipase had got low Km for triacylglycerol in the VLDL compared to adipose tissue lipoprotein lipase. So, tissue specific isoenzymes of lipoprotein lipases are there. So, whenever we have excess VLDL, so most of that triacylglycerol releasing fatty acid it will be taken up by the adipose tissue, and in the adipose tissue that fatty acid is esterified with the glycerol 3-phosphate to make a triacylglycerol. Esterification going on. Where is the esterification going on? It is going on in the endoplasmic reticulum. How do you get this glycerol 3-phosphate in the adipose tissue? You get it from, glucose getting into the adipose tissue. So, it is the glucose. Whenever person is in well-fed condition, glucose getting in using GLUT4 transporter. Remember GLUT4, it is an insulin sensitive transporter, allowing glucose undergoing glycolysis, DHAP to glycerol 3-phosphate by glycerol 3-phosphate dehydrogenase. That's the pathway. So, glycerol 3-phosphate, that's how you get it from DHAP. Remember that. That's an intermediate of glycolysis giving glycerol 3-phosphate. Don't confuse glycerol 3-phosphate with glyceraldehyde 3-phosphate. Okay. So, glycerol 3-phosphate acting as a acceptor of these fatty acids in the esterification process to make triacylglycerol, and that triacylglycerol is loaded in the adipose tissue. So, that's why whenever person consumes glucose, higher glucose, excess glucose under well-fed high carbohydrate diet, remember now what happens? High carbohydrate diet means insulin is more, and insulin is promoting all those enzymes that I have just now explained in the previous minutes. So, glucose all the way to pyruvate, some of its dihydroxyacetone phosphate giving glycerol 3-phosphate, pyruvate to acetyl-CoA, citrate to acetyl-CoA in the cytoplasm, malonyl-CoA fatty acid, and DHAP to glycerol 3-phosphate fatty acid esterified with glycerol 3-phosphate to make triacylglycerol, and that becomes part of VLDL. Okay. Now, cholesterol, acetyl-CoA, mevalonate, FPP, lanosterol, cholesterol ester, and become part of VLDL. So, all this excess glucose carbons, they are all now present as triacylglycerol and cholesterol ester in the VLDL of the liver. Remember, this is what happens to carbohydrate. Consuming excess carbohydrate, it will increase VLDL levels in the blood. Remember, that's how it will contribute to hypertriacylglycerolemia and hypercholesterolemia. How do you decrease hypertriacylglycerolemia and hypercholesterolemia if it is a secondary to lifestyle thing, or if it is a lifestyle issue, if it is not an hereditary condition, if it is just secondary thing, right? So, how do you control that hypertriacylglycerolemia, hypercholesterolemia? You got to control glucose levels, decrease carbohydrate in the food, and so if you decrease that, you are going to decrease VLDL. That means you are going to decrease triglycerides and cholesterol levels in the blood. Very simple treatment. You got to know that. Okay, so all that excess carbohydrates, glucose carbons, they are now present as triacylglycerol and cholesterol ester. Now that VLDL which has triacylglycerol, that is now releasing its fatty acids, and it will go to adipose tissue and store it as triacylglycerol in the adipose tissue. How do you get that? Glycerol 3-phosphate from glucose, DHAP, glycerol 3-phosphate, excess glucose contributing towards that. Fatty acid carbons are basically from glucose there. So, it is now stored as triacylglycerol. So, more adipose tissue means person has consumed more carbohydrates. So, excess carbohydrates will contribute to weight gain, overweight, obesity and also. How do you control obesity? Address this one, decrease glucose, okay, and that can take care of it. Okay, remember that. So, that's how excess carbs, excess carbons, excess energy, now it is stored in the adipose tissue for future needs. Anyway, so that's how VLDLs, triacylglycerol goes on. So, that is what is the role of LPL. What happens to the cholesterol ester which is still there, right? So, we have degraded triacylglycerol. After some point in time, when the size of the VLDL is decreasing, some amount of significant quantity of triglyceride in the VLDL, when it is released as free fatty acid, at that time ApoC2 will go back to HDL, C2 will go back, and when the C2 will go back to HDL molecule, and we refer that VLDL now as IDL, intermediate density lipoprotein. So, it is just a referral, right? It is just VLDL is converted to IDL after release of ApoC2. When the ApoC2 is released, when significant quantity of tags which are there in the VLDL is released as free fatty acids and glycerol. What happened to this glycerol? I have explained what happened to the fatty acid. What happened to the glycerol? Glycerol will go back to the liver, and in the liver, glycerol can have two fate. Glycerol can be converted back into glycerol 3-phosphate undergoing reesterification, or glycerol can go into gluconeogenesis. See this glycerol? Glycerol can go into gluconeogenesis. So, that is the two fate of glycerol in the liver depending on the metabolic condition, fasting, fed and all that. Anyway, so we got ideal there. What happened to the ideal? So, there can be two fates to ideal. Ideal can be going back to the liver. So, entirely it can be taken up by the liver by ApoE receptor. ApoE receptor called as remnant receptors present over liver. They will recognize ApoE which is present over ideal. Remember ideal as ApoB1 and ApoE, okay, important ones. ApoE is recognized by ApoE receptor or remnant receptor, entire molecule is taken back by the liver. That is one fate. Second fate is ideal can be converted into LDL. So, what is the molecule that converts IDL into LDL? So, the conversion of IDL into LDL can be done by two things. One is it can be done by enzyme hepatic lipase enzyme. Hepatic lipase means when IDL is going over the surface of hepatocyte, so hepatic lipase enzyme is going to degrade some of the triacylglycerols present in IDL into free fatty acids again, free fatty acids and glycerol, which will go into the liver. So, tags are further broken down. So, at that time, so there will be release of ApoE. ApoE will go back to HDL. So, that is what we refer it as LDL. That means LDL now purely as this rich in cholesterol ester. So, over a period of time, what happened? This tag and cholesterol ester present in VLDL. Of course, it has vitamin E, vitamin K, right? VLDL is a carrier of vitamin E, vitamin K. So, we are just degrading triacylglycerol here, making IDL still has a tag and cholesterol ester, right? And then the IDL acted by hepatic lipase enzyme releasing only tags, cholesterol is there and ApoE is released. Once ApoE is released after significant quantity of tag is kind of degraded, we re-refer that IDL now as LDL. It's just a referral thing, one after the other. So, VLDL is converted to IDL upon removal of ApoC2, IDL is converted to LDL upon removal of ApoE. How this removal happens? Whenever significant quantities of tags are taken out of VLDL to IDL, IDL to LDL. So, we got LDL now. So, that is one way of converting IDL into LDL. Now, the second way of converting IDL into LDL is there is something called as CETP, cholesterol ester transfer protein. CETP, cholesterol ester transfer protein. This is an enzyme located over HDL molecule. How do you get HDL? So, HDL is actually synthesized by two tissues. One is liver, this is the liver, let me write it down. So, the liver and small intestine. Liver and small intestine has a capacity to synthesize ApoA1. ApoA1 is a major apolipoprotein over HDL. Let me just brief you that in lipoprotein. Major apolipoprotein present over VLDL is ApoB100. Major apolipoprotein present in IDL is ApoB100. Major apolipoprotein present over LDL is ApoB100. Whereas for major apolipoprotein present over HDL is ApoA1. Okay. So, ApoA1, it has got some amount of phospholipid and cholesterol ester. It is a discoidal in shape, disc-like shape coming out of liver and small intestine. In the circulation, what it does, it is going to gain all the other apolipoproteins except ApoBs. ApoB100 and ApoB48 is not there over HDL. Other than those two, every other apolipoproteins are present over HDL. That's why it is a high density lipoprotein. There are a lot of proteins over it. Okay. So, that's an HDL, and HDL, when see this nascent discoidal HDL, it will be converted into HDL 3 initially. Nascent discoidal HDL is converted to HDL 3, and that will be done by enzyme LCAT, lecithin cholesterol acyl transferase enzyme. Nascent discoidal HDL or discoidal HDL is converted to HDL 3 done by LCAT enzyme, lecithin cholesterol acyl transferase. What is this enzyme? Lecithin is phosphatidylcholine present over phospholipid membrane phospholipid, most abundant one, lecithin. Cholesterol, see the cholesterol coming out of the tissue, say peripheral tissues, I will write that here. This is a tissue. Whenever peripheral tissue has the excess cholesterol, this cholesterol will be pumped out of the tissue through ABC A1 and ABC G1 transporter, ABC A1 and ABC G1 transporter, and SRB1, ABC A1, ABC G1, SRB1, that is ATP binding cassette transporter A1, G1, scavenger receptor B1. Tissues, peripheral tissues will secrete that. It's an active mechanism. So, cholesterol, excess cholesterol pumped out of the tissue over HDL molecule. Okay, HDL is in connection with LCAT. There will be LCAT here, this LCAT here. So, LCAT, what it does, the cholesterol that is coming out of tissue will be esterified with the fatty acid to make it as cholesterol ester. It will be converted as cholesterol ester. Cholesterol is converted to cholesterol ester done by LCAT enzyme. So, it is a transfer process. What it transfers? It is going to transfer fatty acid from lecithin. Lecithin is phosphatidylcholine. So, it is taking a fatty acid from that phospholipid, that is phosphatidylcholine. One of the fatty acid is taken from its tail and added to cholesterol to make it as cholesterol ester. So, during that time, what happens to lecithin? Lecithin is converted to lysolecithin, while cholesterol is converted to cholesterol ester. This process is facilitated by LCAT enzyme, and the positive modulator of this LCAT is ApoA1 itself. So, LCAT is positively modulated by ApoA1, whereby cholesterol is converted to cholesterol ester. So, when more and more cholesterol ester is loaded on to HDL molecule, so nascent discoidal HDL will become a spherical HDL3 molecule. Now, cholesterol ester is more in HDL3. What this HDL3 will do? So, HDL3, it will interact with VLDL, IDL and LDL. See, all three of them I have written here. So, what is the function? Cholesterol ester transfer protein. It is there in the name. This is an enzyme which is also present over HDL. HDL has LCAT enzyme, cholesterol ester transfer protein enzyme, all the apolipoproteins except ApoB and ApoB100, and also it has got something called as PON, peroxanase, which is an antioxidant enzyme. Now, what is the role of CETP? Cholesterol ester transfer protein, what it does? It is going to transfer this cholesterol ester to VLDL. In return, is going to take triacylglycerol from this molecule. Okay, it's an exchange process. Similarly, it does with IDL. Cholesterol ester is given and tag is taken. Cholesterol ester is given, and also it does the same thing for with VLDL also. Cholesterol ester is given, tag is taken. Okay, so like this, it is continuously gaining triacylglycerol and giving cholesterol ester. When this process happens, HDL3 will be converted to HDL2, and HDL2 is a big molecule which has cholesterol ester and also it has triacylglycerol, because LCAT continue to work. CETP and LCAT, they are working continuously, loading this HDL to with cholesterol ester and also triacylglycerol. So overall what happens, HDL, nascent discoidal HDL become HDL3 by LCAT enzyme. Now HDL3 become HDL2 by two enzymes, LCAT and CETP. Continuously it is working, both the enzymes are working to convert HDL3 into HDL2. HDL2 is a big molecule. It has lot of cholesterol ester, it has lot of triacylglycerol, and eventually HDL2, it will offload all its content into the liver by SRB1 transporter. SRB1 is interacting with HDL2 and take the content, that cholesterol ester, triglycerides, everything is taken into the liver. Okay, that's HDL metabolism. So overall what HDL has done, HDL has taken excess cholesterol from the peripheral tissue and taken it back to the liver. So this is what is called as reverse cholesterol transport mechanism, which is done by HDL. That's why HDL is referred as a good lipoprotein, because it is not allowing excess cholesterol to be accumulated in the peripheral tissue. Like imagine if it is happening in the arteries leading to atherosclerosis and all that. So HDL is taking back that excess cholesterol to the liver, thereby it can be distributed to other organs, or it is just excreted in the bile as biliary cholesterol. Anyway, so that's what is the function of LCAT, cholesterol ester transport protein. Peroxidase is an antioxidant enzyme, so it is trying to decrease the reactive oxygen molecules, thereby prevent atherosclerosis. Anyway, so with the CETP function with HDL, VLDL is gaining more and more cholesterol, losing triacylglycerol. Same thing is done by IDL, gaining more cholesterol ester, losing triacylglycerol, and same thing is done by LDL, gaining cholesterol ester and losing triacylglycerol. So by the time VLDL is converted to IDL, IDL converted to LDL, even by the action of lipoprotein lipase, losing fatty acid, hepatic lipase, IDL losing fatty acid like triglyceride and only retaining cholesterol ester, and then the CETP function, which is pushing all that cholesterol ester to VLDL, which is becoming IDL, which is becoming cholesterol ester in the LDL. So by the time LDL is made with the action of LPL, hepatic lipase and cholesterol ester transfer protein, LDL is super rich in cholesterol ester. That's why LDL is a cholesterol ester rich molecule. Whenever there is a rise in LDL molecules in the blood, that means there is more cholesterol in the blood, because LDL is rich in cholesterol ester. That's why we will have to reduce LDL levels, keep it less than 100 milligrams. Okay, so what happens with the LDL in the circulation? So LDL is taken up by any tissue that needs cholesterol for its own use. Okay, so how LDL is taken up by the peripheral tissue? Any tissue that needs cholesterol inside the cell, like for its whatever the need of that cell, maybe membrane formation or any special use of cholesterol in specific tissues like gonadal tissue, ovary, testis, for sex hormone formation, or adrenal cortex to make corticosteroids and all that. So anyway, so whichever the tissue it is, so LDL has to be taken up, and any tissue that needs cholesterol, they will express LDL receptor over the surface. And LDL receptor has got 5 domains, domain 1, 2, 3, 4, 5. Domain 1 is called as LDL binding domain, LDL receptor recognition domain. Okay, domain 2, domain 3, domain 4 and domain 5. Domain 5 is there in the cytoplasm, which will interact with a clathrin. It's a clathrin coated, LDL receptor is a clathrin coated receptor. Okay, so when LDL interact with LDL receptor, how it interacts? So ApoB100, which is the only apolipoprotein present over LDL, it is going to interact with LDL receptor, and once that interaction happens, it is internalized. So it is stabilized by clathrin, small molecular weight protein, and once it is internalized, so it is going to go and fuse with the lysosome to become endolysosome, and in the endolysosome the pH will be decreased because the proton pump will increase the pumping of protons inside the lysosome, endolysosome from the cytoplasm. So pH decreases, all the acidic enzymes will be activated, and what they will do, so they are going to break this LDL molecule. So only thing remember, LDL receptor is not destroyed, because there will be hairpin loop kind of thing which will prevent degradation of LDL receptor by this acid stable enzyme. So LDL receptors will come back to the membrane. So every 5 minutes LDL receptor will be recycled out of its 20-minute lifetime. LDL receptor has 20-minute lifetime. In that every 5 minutes it is undergoing a continuous recycling process expressed over the cell membrane. Okay. So whenever, and this hairpin loop thing is actually inhibited by something called as PCSKs. These PCSK9s, so they are inhibiting hairpin loop conformation in the acidic media, and when the hairpin LDL receptor is also be degraded and LDL receptor expression is not there over cell membrane, thereby leading to accumulation of LDL in the circulation. So some people will have higher activity of PCSK9s and they will be prescribed with PCSK9 inhibitors. So what PCSK9 inhibitors will do? They will inhibit PCSK9, thereby they will continue to allow hairpin loop conformation to be formed, thereby LDL receptor can be recycled and make sure that LDL receptors are available over the cell membrane, thereby LDL can bind to LDL receptor and it is taken up by the tissues, thereby decreasing circulating LDL levels or keeping the circulating LDL levels normal. I hope you are getting that. Okay. So now, normally what happens, LDL is broken down into cholesterol, ester released, which is released into cholesterol, amino acids are released, and cholesterol is converted to cholesterol ester by ACAT enzyme, acyl cholesterol acyl transferase, and the cholesterol ester either become part of the membrane or it can go into some other cellular use of cholesterol. And note that whenever there is excess cholesterol inside the cell, there will be decreased expression of LDL receptor over the surface, because already cell has sufficient cholesterol. So the cholesterol itself will have a negative effect on the gene that is coding for LDL receptor, thereby LDL receptor gene is not activated, that means LDL receptor protein is not synthesized, thereby decreasing the LDL receptor expression over the cell membrane. So, and also excess cholesterol inside the cell will have a negative effect on FMGE reductase enzyme. So, all this negative effect will go on. So, if there is a less cholesterol inside the cell, more LDL receptor over the surface. If there is more cholesterol inside the cell, less LDL receptors over the cell membrane. Make sure you remember that. Okay, and that's what happens to LDL in the circulation. So, I have covered VLDL coming from the liver, how it is made, VLDL to IDL, IDL to LDL, and then I have explained HDL metabolism. Only the lipoprotein that is left here is chylomicron metabolism. Alright, so how chylomicrons are made? So in order to understand chylomicron, let me briefly explain you digestion and absorption of food. Major macromolecules present in the food are carbohydrates, proteins and lipids. So carbohydrate digestion by carbohydrate digestive enzymes, you know, amylase, salivary amylase, pancreatic amylase. It will break alpha 1,4 bond of the starch and glycogen, release glucose molecules, right? So we get glucose. Predominant carbohydrate is glucose and minor carbohydrates like galactose and fructose. So glucose gets into the portal circulation, taken up by the tissues. So uptake is done by GLUT5 transporter over the brush border epithelium. SGLT1 transporter is there over brush border epithelium. SGLT1 transporter, that is sodium dependent glucose transporter, is a secondary active transporter because sodium potassium pump has to go on to make sure sodium gradient is maintained, SGLT1 is working, all that information for glucose absorption. GLUT4 transporter is a facilitated transporter, works by the concentration gradient. It will allow glucose, galactose, fructose to go in, whereas SGLT1 allows only glucose and galactose. And once more and more glucose, galactose, fructose is there in the intestinal epithelial cell cytoplasm, it will go into the portal circulation by GLUT2 transporter, and that's how glucose, galactose, fructose goes into the portal circulation. From there it is taken up by the liver and from there all the other organs. So that's what it is. Coming to proteins, and also before that carbohydrate, I have told you two carbohydrate digestive enzymes, salivary amylase and pancreatic amylase, which will break alpha 1,4 bond, glycosidic bond. What about maltose, isomaltose, oligosaccharides, lactose, sucrose, which enzymes will break? So there are brush border epithelium enzymes which will take care of maltase enzyme is there, which will break alpha 1,4 bond, and then isomaltase dextrinase enzyme complex, you know, isomaltase breaks alpha 1,6 bond present in isomaltose, and then the lactase enzyme breaks beta 1,4 bond, sucrase enzyme which will break alpha 1,2 bond of sucrose. Lactase enzyme breaks lactose into glucose and galactose by breaking beta 1,4 bond. Sucrase enzyme breaks sucrose into glucose and fructose by breaking alpha 1,2 bond. And then the alpha glucosidase enzyme will work on the oligosaccharides, break alpha 1,4 bond one at a time from the non-reducing end. Okay, so these are some of the enzymes, and acarabose is the one that will inhibit alpha glucosidase enzyme. Anyway, so that's carbohydrate digestive enzymes. Coming to proteins, and before that, GLUT transporters, GLUTs, like glucose transporter, there are 12 of them, like 1, 2, 3, 4, 5, and then all the way to 12. Most important one in that is the GLUT4 transporter. Remember, GLUT4 transporter is an insulin sensitive transporter which are expressed in the over the membrane of adipose tissue, skeletal muscle and cardiac tissue. Remember, this is the only insulin sensitive transporter, whereas GLUT1 and GLUT3 transporters are low Km transporter, that means high affinity glucose transporter. GLUT1 is expressed over red blood cell and all other barriers like blood brain barrier, and wherever you see barriers, that's GLUT1. GLUT3 transporter is there in the neuronal tissue, brain. Okay, both are GLUT1 and GLUT3, they are low Km transporters, whereas GLUT4 is a insulin sensitive transporter. GLUT2 is a high Km and high Vmax transporter present in the liver, kidney, beta cells of pancreas, and also in the basal side of small intestinal epithelial cell. Anyway, so coming to the proteins. Proteins are digested by concerted action of so many enzymes. So with the specificity of their, each one of them are specific for some of the bonds. So what are those? Protein undergoing denaturation in the stomach by the acidic media. So it will open up like quaternary structure, tertiary, secondary structure opens up because they are non-covalent interaction. Now the primary structure with the peptide bond, it will be clipped by pepsin in the stomach, which is activated by hydrochloric acid. So pepsin is more effective on the carboxyl side of, which is an endopeptidase, means breaks within the polypeptide chain. It is active on the carboxyl side of aromatic and acidic amino acids. Remember that. What all aromatic amino acids? Tyrosine, tryptophan, phenylalanine. Acidic amino acids, that is aspartate and glutamate. Carboxyl side, you know, amino side on the left side, carboxyl side on the right side. Remember that. And then you got oligopeptides and all that. So they will go into the duodenum, and in the duodenum these pancreatic enzymes will come. So these pancreatic enzymes, they are in pro form, like zymogen form. Zymogen form of pancreatic enzymes need to be activated into their active form, and that is done by enterokinase or enteropeptidase enzyme present over brush border epithelium. So it is going to initially convert trypsinogen into trypsin, and that trypsin converts further trypsinogen into trypsin, chymotrypsinogen into chymotrypsin, proelastase into elastase, procarboxypeptidase A into carboxypeptidase A, procarboxypeptidase B into carboxypeptidase B, and all these active enzymes now, based on their specificities, they are going to work on all the partially digested polypeptide chain coming from the stomach. You got to remember the specificity of those enzymes. Okay, so now trypsin, it's basically acting on the carboxyl side of basic amino acids like arginine, lysine, and then the chymotrypsin hydrophobic amino acids and aromatic and hydrophobic amino acids specifically. Elastase acts on the small side chain amino acids like alanine, glycine, kind of like that. And then the procarboxypeptidase A, procarboxypeptidase B, their action is similar to trypsin and chymotrypsin respectively. So, and then we have, these are all endopeptidases, whereas the like trypsin, chymotrypsin, elastase, pepsin, these are endopeptidases. Carboxypeptidases are exopeptidases, it is there in the name. So, carboxyl side of the polypeptide chain they will remove, and there is aminopeptidase. So, proaminopeptidase activated into aminopeptidase, amino side it will remove. Like this concerted action of all these enzymes. So, we are going to get di and amino acids and dipeptides, and dipeptides, amino acids taken inside the epithelial cell, dipeptidase enzymes inside. So, they are going to make ultimately into amino acids, and amino acids are going into portal circulation. That's how amino acids go there. Now, coming to the lipids. Okay, so what are the lipid digestive enzymes? So, we have lingual lipase and gastric lipases. These are acid stable enzymes. So, they are going to work on the triacylglycerol, which have got short and medium chain fatty acids, And then triacylglycerols, basically lipid in the diet is 90% is triacylglycerol. Then we have cholesterol ester, phospholipid, vitamin A, D, Ks and all that. So in the duodenum what happens? So pancreatic lipase will come. So this pancreatic lipase along with the co-lipase, so it is going to work on remaining triacylglycerol, and then we have cholesterol esterase, pancreatic cholesterol esterase, pancreatic phospholipase A2, all of them need to work. So there is a requirement for lipid digestion. What is the requirement? Requirement is there must be sufficient bile acids and bile salts available, because in the presence of bile acids and bile salts, all these hydrophobic lipids are basically coated by bile acids and bile salts, and intestine contraction has to go on to break the lipid molecule into simple small molecules called micelles. So breakdown of a big lipid, hydrophobic lipid, into small lipid particles is referred as emulsification process, and for emulsification there is a need for bile acids and bile salts. Intestine has to contract. Okay, so once this happens, the micelles which are coated by bile acids and bile salts with the triacylglycerol, cholesterol ester, phospholipid, vitamin A, D, K, all that is there in the center of this molecule. Now these molecules are acted by pancreatic lipase along with the co-lipase. They both are partners, and then the pancreatic cholesterol esterase and pancreatic phospholipase A2. So what pancreatic lipase co-lipase does? It's going to take care of triacylglycerol. It will take away short, medium, long chain fatty acids from first and the third carbon. Second carbon it doesn't touch. Okay, so that's why after the action of pancreatic lipase with co-lipase, we are going to get triglycerides broken down into two individual free fatty acids and one molecule of 2-monosylglycerol. Okay, and then the cholesterol esterase enzyme break cholesterol ester into cholesterol and fatty acid. Phospholipase A2 break phospholipid into fatty acid and lysophospholipid. All of them, they are in association with the bile acids and bile salts along with vitamin A, D, Ks, and this is referred as a mixed micelle. So micelles are converted into mixed micelles by the action of linguolipase, gastric lipase, pancreatic co-lipase, pancreatic cholesterol esterase, pancreatic lysophospholipase A2. So now we have mixed micelle with the digested content. What all the digested content? We have free fatty acid, 2-monosylglycerol, cholesterol, lysophospholipid, and then of course vitamin A, D, K. All of them need to undergo simple diffusion process. So most of them will undergo simple diffusion in the duodenal and jejunal brush border epithelium, but only thing is there is a watery layer on the surface of brush border epithelium, and bile acids and bile salts will go and interact with the water layer over the brush border epithelium and take it away, and then now the simple diffusion of free fatty acids, 2-monosylglycerol and lysophospholipid, all these things, vitamin A, D, Ks, they will go on. Only issue here is the cholesterol, which is a big molecule. It needs a transporter, and that is called NPC1L1, Niemann-Pick C1 like one transporter, which will allow transport of cholesterol inside. It's an active process, so ATP binding cassette transporter type of thing. Okay. Now there is a drug called ezetimibe which will inhibit NPC1L1, and the orlistat will inhibit pancreatic lipase and gastric lipase enzyme. That's how they are used in treatment of hypercholesterolemia or obesity. Orlistat is used as anti-obesity drug. Now, once all these things are inside the enterocytes by simple simple diffusion and by NPC1L1, so once these things are inside, like free fatty acid esterified with the glycerol 3-phosphate to make triacylglycerol, and then the free fatty acid esterified with 2-monosylglycerol to make triacylglycerol. Cholesterol is esterified to make cholesterol ester, lysophospholipid esterified with the fatty acid to become phospholipid, and vitamin A, D, K. All of them now will be loaded onto ApoB48, and this ApoB48 is actually coded by ApoB100 gene. You get to ApoB100 mRNA, but it will undergo mRNA editing in the intestine. Okay, and that is done by an enzyme called cytidine deaminase, and this cytidine deaminase is going to convert a sense codon which is coding for glutamine into a non-sense codon. Okay, so this is how with the mRNA editing process, so ApoB48 is synthesized. Remember that part, cytidine deaminase, remember that it is expressed in the intestinal cell. Now with the B48, all these things are loaded onto B48, and that makes a nascent chylomicron, which is a big molecule. okay, and this is all going on in the esterification is going on in the smooth ER, B48 is there in the rough ER. So lipids from the smooth ER are transported to B48 present or rough ER, done mediated by MTP, microsomal triacylglycerol transfer protein, MTP. If there is a MTP gene is mutated, it will give rise to a disease called A beta lipoproteinemia. Remember that. Okay, so this nascent chylomicron, I have a video on this lipoprotein metabolism. If you didn't get lipoprotein metabolism, please go over that video. It is there in YouTube. Okay, the link for that is given below. So now, once this nascent chylomicron enters into the circulation, so just like any other nascent like VLDL molecule, it's going to gain important apolipoproteins from HDL. So it's going to gain ApoC to ApoE, important ones. Other things I have not mentioned. So we have B48, ApoC to ApoE over chylomicrons, and note that chylomicrons are big, so they go into lymphatics first and then enter into the systemic circulation. That's when they are going to get ApoC2 and ApoE. Okay, otherwise in the lymphatics it is still ApoB48. Remember that. So once it is matured after getting C2 and E in the circulation from HDL, so lipoprotein lipase enzyme LPL, just like how I have explained with VLDL. So LPL, which is attached to the endothelium of the blood vessel with heparan sulfate, it is activated by ApoC2, as I have already explained to you. LPL, once it is activated, it is going to degrade triacylglycerol. 90% of chylomicron is triacylglycerol. It is degraded into free fatty acid and glycerol, done by lipoprotein lipase. And this is all an exogenous triacylglycerol coming from the food, as you have seen. So glycerol will be taken up by the adipose tissue. If it is, adipose tissue LPL is a high Km, high Vmax LPL. Of course, triglycerides can also be taken by peripheral tissues, cardiac tissue and all that, but most of it will go to the adipose tissue and is stored there as triacylglycerol, white depot fat. Glycerol will go back to the liver and it can undergo esterification, glycerol 3-phosphate and re-esterification process in the liver. Okay. Now, as this more and more triglyceride is degraded coming in the chylomicron, so at some point in time when significant degradation goes on, at that time ApoC2 will go back to HDL. So now we have only B48 and ApoE and very less triglyceride and vitamin A, D, Ks, phospholipid, cholesterol, all that is there. And that molecule we call it as chylomicron remnant. And this chylomicron remnant has B48 and ApoE. And the chylomicron remnant is taken up by the liver, because liver is going to recognize that ApoE by remnant receptor or ApoE receptor recognizes this ApoE, entire molecule is internalized. That's how cholesterol, phospholipid, vitamin A, D, Ks along with some of the triglycerides which are not degraded by LPL, they all will be ended up in liver, and that's what is the chylomicron metabolism. So what happens if there is a defect in LPL, lipoprotein lipase deficiency? It will lead to elevated chylomicron and also it will lead to elevated VLDL. Remember VLDL to ideal, you need lipoprotein lipase. So there will be elevation of VLDL, elevation of chylomicron. VLDL and chylomicron both are rich in triacylglycerol. That's why it will give rise to familial hypertriacylglycerolemia. Okay. That's one of the lipoprotein, that's because of lipoprotein lipase deficiency, hyperlipoproteinemia. Okay. So what if there is a deficiency of LCAT? LCAT deficiency it can lead to decrease in the conversion of discoidal nascent HDL into HDL. So there will be low levels of HDL. If there is a deficiency of ABCA1, ABCG1 transporter, it can give rise to cytosterolemia type of condition, and then Tangier disease can happen. So and then LDL receptor deficiency, it can lead to familial hypercholesterol. Alright, so as you see here, the chylomicron and VLDL is super rich in triacylglycerol. So what happens whenever a person is in fed condition? So most of this triacylglycerol ultimately it ends up in adipose tissue, because the free fatty acids from chylomicron degradation and VLDL degradation will be going into adipose tissue and re-esterified there as triacylglycerol. So what happens to this triacylglycerol? So whenever person is in fasting condition or when the person is exercising, so there will be release of glucagon, epinephrine. So glucagon binding to glucagon receptor, epinephrine binding to beta to adrenergic receptor, which are G protein coupled receptor, So they will activate adenylate cyclase, cyclic AMP elevated, and ultimately activating protein kinase A enzyme. And this protein kinase A is going to activate hormone sensitive lipase enzyme. So hormone sensitive lipase, once it is activated, what it does, it's going to break triacyl glycerol into free fatty acid and glycerol molecule, and glycerol will go to the liver and it will undergo gluconeogenesis process, because person is in fasting and exercise and all that. So gluconeogenesis of glycerol is glycerol converted to glycerol 3-phosphate, dihydroxyacetone phosphate, go all the way to glucose. We have already seen that. So that's the fate of that glycerol. So what happens to the free fatty acid? Free fatty acid is flooding into the blood, especially long chain fatty acids. They are not soluble in the blood, whereas short and medium chain fatty acid, they are soluble, so they don't need any transporter. Whereas long chain fatty acid, it needs a transporter, and albumin in the blood act as a transporter of long chain fatty acid. So this long chain fatty acid bound albumin carried in the blood going to the peripheral tissues, including in the liver. So what happens? So the fatty acids undergo free diffusion through the cytoplasm, while albumin goes back into the blood, and fatty acids need to go inside the matrix of mitochondria to undergo beta oxidation into acetyl-CoA, and that is a problem there. So because the mitochondrial inner mitochondrial membrane especially is not allowing long chain fatty acids directly, so we need a transporter, and the transporter for that is a carnitine shuttle mechanism. And this carnitine shuttle mechanism, where carnitine is involved in the transport of long chain fatty acids into the matrix of mitochondria. In the carnitine shuttle mechanism, you will have to remember that outer mitochondrial membrane, we have acyl-CoA synthetase enzymes where fatty acid is activated by using 2 ATPs, and then the carnitine palmityl transferase 1 is there in the outer mitochondrial membrane, which is converting fatty acyl-CoA into fatty acyl carnitine using carnitine there, and that is called CPT1 or CAT1. And then in the inner mitochondrial membrane, we have carnitine acyl carnitine translocase enzyme, and then the CPT2 enzyme is there, carnitine palmityl transferase 2, which is also called as CAT2, that is carnitine acyl carnitine translocase, this carnitine acyl transferase 2 enzyme. So with the help of these enzymes, so with the help of carnitine, of course, carnitine is a dipeptide coming from lysine and methionine, predominantly synthesized in the liver, and liver issues, liver problems will lead to decrease in carnitine. Kidney issues like renal filtration problem can also lead to carnitine deficiency. Lysine methionine deficiency will lead to carnitine deficiency. That means essential amino acid deficiency will lead to carnitine deficiency. So liver diseases, nutritional deficiency of essential amino acids, kidney diseases, there will be carnitine deficiency. That means long chain fatty acids are not undergoing beta oxidation. Those things you'll have to remember. CPT1, CPT2 enzymes can also be deficient because of mutation in a gene coding for CPT1 or CPT2 enzyme. So that will also lead to disorders associated with carnitine, okay, carnitine deficiency issues where beta oxidation of fatty acids, long chain fatty acids don't go on, okay, that kind of. So I have a video on that, you can watch that video if you didn't get that concept before. Now what happens in the matrix of mitochondria? So fatty acids undergoing oxidation, especially at the beta carbon, that is the third carbon in a fatty acid is a beta carbon, second carbon is alpha, third carbon is beta. Beta carbon undergoes oxidation, and there are 4 reactions there, and the first reaction and the third reaction is oxidation reaction there, where first reaction is acyl-CoA dehydrogenase enzyme, where FAD is getting into the reaction, FADH2 comes out, and the third reaction is beta hydroxyacyl-CoA dehydrogenase reaction, where NAD getting in and NADH plus gets out. So in each beta means at the end of one beta oxidation spiral, that is number 1, 2, 3, 4 reaction, there will be release of one FADH2, one NADH plus, and your long chain fatty acid or any fatty acid. In fact, it will break into 2 carbon is released as acetyl-CoA, and rest of the fatty acid chain will go back to the first reaction. So like this, every beta oxidation spiral, there will be release of one acetyl-CoA, one FADH and one NADH. You know, one FADH is 1.5 ATP, one NADH is 2.5 ATP. Total you get 4 ATPs from NADH and FADH, and one acetyl-CoA going into acetyl TCA cycle, you know you get 10 ATP. We have calculated that 10 ATP. So in one beta oxidation spiral, effectively we get 14 ATPs, okay, and that's how you can calculate total number of net number of ATPs coming from oxidation of a fatty acid. Anyway, so that video is also there in the in my channel. You can watch it later. So ultimately, what I am saying here is fatty acids are undergoing oxidation to release acetyl-CoA. Know that this can go on in any tissues, okay, except in the brain. Brain and red blood cells don't conduct beta oxidation of fatty acid. Now, acetyl-CoA, what happens to the acetyl-CoA? Depending on the tissue type, acetyl-CoA can go into TCA cycle for the energy needs of the tissue, and especially in the liver, acetyl-CoA can be diverted into ketone body formation, especially in the mitochondria of the liver. Acetyl-CoA diverted into HMG-CoA formation, acetyl-CoA combining with another acetyl-CoA to make acetoacetyl-CoA, that will combine with one more acetyl-CoA in the matrix of mitochondria to make HMG-CoA, and then HMG-CoA breaks by HMG-CoA enzyme and you get acetoacetate and acetyl-CoA. And acetoacetate is the first ketone body, and that acetoacetate, most of it is diverted into beta hydroxybutyrate or 3-hydroxybutyrate done by beta hydroxybutyrate dehydrogenase enzyme. In this reaction, NADH is used and NAD plus gets out. It's a regeneration of NAD plus for beta oxidation process. That's why most of the acetoacetate is diverted into beta hydroxybutyrate. In fact, for every 4 to 5 beta hydroxybutyrate in the blood, you're going to find one acetoacetate. And when this reaction, that is conversion of acetoacetate into, let me write it down. So ketone bodies are, first is acetoacetate, which is diverted into beta hydroxybutyrate, and other is acetone. Okay, so these three are the ketone bodies. Actually, the first ketone body, primary ketone body is acetoacetate, and that's a true ketone body. Acetone is also true ketone body, but beta hydroxybutyrate is a derivative of ketone body, that is acetoacetate converted into beta hydroxybutyrate. Most of the acetoacetate in the blood is actually in the form of beta hydroxybutyrate. Okay. That is because to regenerate NAD plus, which is required for beta oxidation of fatty acid and even for TCA cycle. Now, when this acetoacetate conversion into beta hydroxybutyrate is saturated, at that time some of the acetoacetate is diverted into acetone, which is a volatile molecule. Person with ketoacidosis actually expires this acetone in the breath, and the breath will have a fruity odor or the acetone smell to the breath, and that will indicate severity of the ketoacidosis. Remember that. Okay, so when the urine test is done, so generally we do acetoacetate and beta hydroxybutyrate because acetone is a volatile molecule. Remember that part. So presence of acetone in the breath indicates severity of the severe ketoacidosis. Anyway, so that's about beta oxidation of fatty acid. That's the fate of triacylglycerol that is stored in the adipose tissue. And this completes most of the lipid metabolism. So far I have completed carbohydrate metabolism here and connected that with the lipid metabolism. So including like I have explained you, fatty acid synthesis, cholesterol synthesis, connected that with lipoprotein metabolism, and then also I have explained briefly on carbohydrate, protein and lipid digestion and absorption process, and some of the disorders I have mentioned there. Now let me take you into amino acid metabolism. So amino acids, there are 20 standard amino acids, and these 20 standard amino acids, when we are taken in the food like proteins converted to amino acids can go into new protein synthesis. That's one thing, very primary thing there. Now what happens to excess amino acid? Whenever person consumes excess proteins which are giving rise to excess amino acids beyond the need of a new protein that is synthesized in the cell, so these amino acids can undergo breakdown process. Basically, amino acid has got amino group and the carbon chain. Okay, so the amino group, primary amino group attached to the alpha carbon, it is undergoing, amino acids will undergo transamination. Out of 20 standard amino acid, 17 of them will undergo a transamination process, except proline, hydroxyproline, and then threonine and lysine, which don't undergo transamination process. Okay, so other than these 3 amino acids, because hydroxyproline is a derivative of proline, so 17 of them will undergo transamination process. So their alpha carbon is basically given to alpha ketoglutarate, which will become glutamate. I have a video on transamination process. In this video I am not going into detail of that, so please watch that video if you didn't get that particular concept, transamination, oxidative deamination and all that process. Anyway, so alpha carbon amino group is taken care. Now the carbon skeleton of an amino acid is released as a keto acid, like alanine converted to pyruvate, aspartate converted to oxaloacetate at the end of transamination. Like this, corresponding keto acids will be released, and what will happen to that keto acid, corresponding keto acid. So pyruvate from alanine, what happens to the pyruvate? Pyruvate can go into gluconeogenesis, pyruvate, once you get alanine, pyruvate, it can go into glucose formation. So alanine is a glucogenic amino acid. Like this, 18 out of 20 standard amino acid except leucine and lysine. So all of them, they can undergo gluconeogenesis. That's what I have written here. So we have 18 amino acids that can undergo glucose, that can give glucose formation. So except leucine and lysine, which are purely ketogenic. So you can remember that as leucine-lyse. So leucine and lysine. Other than that, all other amino acids can make glucose, one or the other way, getting into gluconeogenic pathway. Remember that. Now, out of that 18 amino acids which are glucogenic, some of them, they are both glucogenic and ketogenic. You should remember that. And those are 3 aromatic amino acids, that is phenylalanine, tyrosine and tryptophan, and isoleucine and threonine. So that basically 5 amino acids, they can make both glucose and ketone bodies. That is 3 aromatic amino acids, phenylalanine, tyrosine, tryptophan, and isoleucine, threonine. Remember that. Make a mnemonic and remember that. Okay. So that's about glucogenic and ketogenic, both glucogenic and ketogenic amino acids. So 18 amino acids can make glucose. 2 amino acids make ketone bodies, and 5 amino acids make both glucose and ketone body. Remember that. Okay. Now, so a little bit about a nitrogen balance. So some of the amino acids, they are synthesized in our body, which are referred as non-essential amino acids. Some amino acids are not synthesized in our body, which are essential amino acids. Some amino acids are synthesized to a certain extent in our body, which are referred as semi-essential amino acids. Like the best example for that is arginine, and some books will mention even histidine as a semi-essential amino acid. Even some resources will mention cysteine and tyrosine also as a semi-essential amino acid. But best example, if you have 4 of them like arginine, histidine, cysteine, tyrosine, choose arginine as your first choice. If arginine is not there, if the question is about semi-essential amino acid, go for histidine. If histidine is not there, still the question is about semi-essential amino acid, go for cysteine if it is there, or tyrosine if it is there. So this is what about, this is about those confusing type of question. You can take care of it. Now, why did I say about essential, semi-essential and non-essential amino acid is because the understanding nitrogen balance. What is nitrogen balance? Nitrogen balance is amount of nitrogen that is getting into our body should be equal to the amount of nitrogen that is going out of our body. So, okay. So, what is the source of nitrogen that is getting into our body? It is the protein, because protein it is a polypeptide chain. It is a connection of amino acids with one another, and amino acid has got alpha carbon attached with the amino group, primary amino group, primary carboxyl group, H, and the side chain, and even some amino acids have nitrogen in the side chain. But every amino acid has got amino group attached to the alpha carbon, and that is the source of nitrogen. So the amount of protein that is getting into our body, which is in the form of protein, should be equal to the amount of nitrogen that is going out of our body. How nitrogen goes out of our body? I told you during transamination what happens. Say when alanine is undergoing transamination, as I told you, by alanine transaminase. So let me write it down. So there is alpha ketoglutarate, which is converted to glutamate. Okay. So and that means the amino group of alanine is now present in glutamate. It is transferred, and the carbon part of alanine is now present in pyruvate. So that's how the amino group is given. So it is there in the glutamate. What happens to the glutamate? Glutamate will be converted to glutamine. That is done by glutamine synthetase enzyme. That means one more ammonium ion is loaded onto this, and this glutamine, when it goes to the liver, it will be reversed into glutamate, releasing ammonium ion. That is done by glutaminase enzyme, and this ammonium ion will go into urea cycle. Okay, glutamate undergoes glutamate dehydrogenase reaction, becoming alpha ketoglutarate, releasing one more ammonium ion, and that also goes into urea cycle, because glutaminase and glutamate dehydrogenase enzymes, so they are present in the matrix of mitochondria, and urea cycle is also going on in the matrix of mitochondria. So let me briefly go over urea cycle. So in urea cycle what happens? So let me see if I have written as it is there here. So here it is. See, amino acid, example I have written alanine, amino acid alanine, example, and alpha keto acid, that is alpha ketoglutarate is the common alpha keto acid which is getting into transamination. So, amino group of alanine is given to alpha ketoglutarate, that's when alpha ketoglutarate becomes glutamate. Okay, that means amino group of alanine, that is NH3 here, will become a part of glutamate. Okay, and the carbons of alanine will become alpha keto acid, that is pyruvate. Okay, now your amino group is in glutamate. Glutamate is converted to glutamine, as I told you already. See this, it takes ammonium ion becoming glutamine by glutamine synthetase enzyme. So that glutamine getting into the blood, so it is taken up by the liver, it is taken up by the kidney, it is taken up by the small intestine. So especially in the liver and kidney, what happens? So there is a glutaminase enzyme in the matrix of mitochondria, glutaminase, and this glutaminase is going to release this ammonium ion, and that ammonium ion, it goes into urea cycle. Okay, so that's one thing. Now glutamine is converted to glutamate. Now this glutamate will undergo, it can undergo two fate. One is it can undergo glutamate dehydrogenase reaction in the matrix of mitochondria, releasing one more ammonium ion, and that goes into the urea cycle. So other fate is glutamate can undergo transamination reaction catalyzed by AST, aspartate transaminase, where oxaloacetate is getting the amino group from glutamate and becoming aspartate. So the amino group of glutamate is there in the aspartate now, and glutamate is carbon skeleton of glutamate now released as alpha ketoglutarate. Now that aspartate which has got amino group, primary amino group, is, it can go into urea cycle also. Okay, that means ammonium ion directly going into the urea cycle, aspartate also going into the urea cycle. Let's look into that. So ammonium ion combining with carbon dioxide, bicarbonate, as carbon dioxide coming in as bicarbonate, using 2 ATP will become carbamyl phosphate, and that is done by enzyme carbamyl phosphate synthetase 1 enzyme, CPS1 enzyme, which is one of the regulated, most regulated enzyme in the urea cycle, rate determining step in the urea cycle, going on in the matrix of mitochondria, first reaction of urea cycle. And remember, NAG, N-acetyl glutamate, is a obligate activator of carbamyl phosphate synthetase 1, and that N-acetyl glutamate is in turn is synthesized by an enzyme called N-acetyl glutamate synthase enzyme by using acetyl-CoA and glutamate, and arginine is the positive modulator of NAG synthase, N-acetyl glutamate synthase. Arginine, whenever there is excess arginine in the liver or in the liver, basically indicating that protein synthesis is not going on, because if the protein synthesis is going on, arginine might have been used. Now if arginine is more, that means cell don't want protein, means no longer conducting protein synthesis, excess amino acids are available. So arginine acts as a NAG activator of NAG synthase, making NAG, N-acetyl glutamate, combining acetyl-CoA with the glutamate, and that NAG is a positive activator of carbamyl phosphate synthetase. In fact, it is an obligate activator of carbamyl phosphate synthetase 1 enzyme. So in our body there are two obligate activators. One is NAG on CPS1, other is acetyl-CoA. Here it is, acetyl-CoA on pyruvate carboxylase enzyme. These two are the obligate activators on enzymes. Okay, so once you get carbamyl phosphate from carbamyl phosphate synthetase 1 enzyme, this carbamyl phosphate condenses with ornithine to make citrulline, and that is done by ornithine transcarbamylase enzyme, and this is also present in the matrix of mitochondria. So first and second reaction of urea cycle goes on in the matrix of mitochondria. Ornithine enters into the matrix of mitochondria from the cytoplasm by ornithine transporter, which can be defective in a syndrome called HHH syndrome. Okay, so now with the condensation of ornithine with carbamyl phosphate, you get citrulline, and this citrulline will get out of mitochondria into the cytoplasm as ornithine is coming in. So it is a ornithine citrulline antiporter system, so which can be defective in HHH syndrome. Okay, now citrulline, it will condense with aspartate to make argininosuccinate, and that's the third reaction of urea cycle going on in the cytoplasm. Okay, and this reaction needs ATP, which is converted into AMP. So ATP basically 2 ATPs are used in this reaction. So 2 ATPs are used in the first reaction, 2 ATPs are used in the third reaction in the urea cycle. So in the formation of urea there are total 4 ATPs being used. So argininosuccinate synthetase enzyme is condensing arginine with aspartate with citrulline to make argininosuccinate. Now the argininosuccinate will break into arginine and fumarate by argininosuccinate lyase enzyme. And now arginine will undergo it, and arginine can have two fate in the liver. It can go into protein synthesis, and if the person is in not positive nitrogen balance, means when protein synthesis is not going on, arginine goes into arginase enzyme which will break arginine into urea and ornithine. So basically the guanidinium group that is there in the arginine, it will be released as urea. So NH2CO NH2 there. So it will arginine breaks into ornithine and urea. Okay. Now that urea is secreted by the liver into the blood, and the kidney is going to filter it and put that into urine, and that's how urea goes out of our body through kidney into the urine. Okay, that's what happens to the glutamine which is going into the liver. What about kidney taking up glutamine? Glutamine in the kidney, it undergoes glutaminase reaction similar to what I have explained you, releasing ammonium ion, and that goes out into the urine directly. And then the glutamate undergoes glutamate dehydrogenase reaction, releasing ammonium ion, that goes into the urine directly. So basically kidney acts as a second line of defense against hyperammonemia, whereas liver act as a first line of defense against preventing hyperammonemia there. Anyway, so this is what is the urea cycle. So argininosuccinate breaks into arginine and fumarate. Arginine goes into arginase reaction, making ornithine, regenerating ornithine. Ornithine goes back into the urea is secreted. Now the fumarate coming out of argininosuccinate, it can continue, it can go to fumarate to malate, malate to oxaloacetate, oxaloacetate to aspartate. So that's like a Krebs cycle continued. So it is sometimes referred as Krebs bicycle, combination of urea cycle and TCA cycle connection there. Anyway, so that's how the urea cycle. So there can be defect in the urea cycle enzymes, like first enzyme deficiency, like CPS1 enzyme deficiency lead to hyperammonemia type 1, severe condition, excess ammonium ion in the blood, and then excess glutamine. Always remember, whenever there is hyperammonemia, there is excess glutamine in the blood. And then the number 2, that is ornithine transcarbamylase enzyme deficiency, will lead to OTC deficiency, ornithine transcarbamylase enzyme deficiency, and that will lead to orotic aciduria, one of the important finding in OTC deficiency. Along with increase in glutamine, increase in ammonium ion, look for orotic aciduria, because carbamyl phosphate, which is elevated in OTC deficiency, comes out of mitochondria into the cytoplasm, go into pyrimidine biosynthesis, where it leads to excess orotate. Orotate goes out in the blood into the urine, leading to orotic aciduria. Okay, and then we have deficiency of argininosuccinate synthetase enzyme giving rise to citrullinemia, and then the deficiency of argininosuccinate lyase enzyme giving rise to argininosuccinic aciduria, and deficiency of arginase enzyme giving rise to hyperargininemia. So as you go from number 1 to number 5, enzyme deficiency in urea cycle, the severity of hyperammonemia actually decreases. Okay, that's little bit about urea cycle disorders there. Now let me take you into, so I was there with nitrogen balance. I'm sorry about that, I just taken you all the way towards transamination. So nitrogen balance, it is the amount of nitrogen that is coming into the body should be equal to the amount of urea that is going out of our body. This urea here, you look at urea. Urea has NH2CO NH2, that means it has 2 nitrogens there. So nitrogen is going out in the form of urea. It's the major nitrogen excretory product of our body, urea in our body. Okay, so protein coming in, nitrogen coming in the form of protein should be equal to the nitrogen that is going out of our body in the urine. That is what is called as nitrogen balance. What is positive nitrogen balance? Positive nitrogen balance means if the amount of nitrogen that is coming into the body in the form of protein, if it is more than the urea that is going out of our body, that means nitrogen is retained in the form of new proteins in our body. That is what is called as positive nitrogen balance. Example like pregnancy, where new proteins are synthesized for the growing fetus inside, or childhood where growth and development is going on, or when the person is gaining weight, like muscle building, person is doing a weight training exercise, muscle building, bodybuilders, they go into positive nitrogen balance. Or person recovering from illness, so during illness, catabolic condition where proteins are degraded, skeletal muscle protein degraded, now that protein rebuild process going on, that is a positive nitrogen balance condition. So these are positive nitrogen balance. Now coming to the negative nitrogen balance. Negative nitrogen balance is when the amount of protein that is coming into the body is much less than the amount of urea that is going out of our body. It's just a negative thing. More nitrogen going out than nitrogen that is coming in. That means more and more proteins that are there in the body is broken down and making urea. It's a catabolic condition. So negative nitrogen balance is seen in catabolic condition like chronic disorders, cancer, cachexia, then the essential amino acid deficiency, or it may be starvation like kwashiorkor, protein deficiency, then it can be severe illness, infection, debilitating disorders, burns, septicemia. These are some of the negative nitrogen balance conditions that you must remember. Okay, so now let me then the essential amino acids you can remember as PVT, Timhol, Pyruvate, Timhol, where HALL. A is arginine, H is histidine. So I told you already, arginine is the most semi-essential amino acid. Partly you can add histidine there. Now let me briefly take you into some of the important amino acid metabolism which can be asked in the exam. So first let me take you into tyrosine or say phenylalanine, metabolism of phenylalanine other than going into protein synthesis. So the phenylalanine undergoes hydroxylation reaction catalyzed by phenylalanine hydroxylase enzyme which needs BH4 tetrahydrobiopterin. Phenylalanine hydroxylase converting phenylalanine into tyrosine, where hydroxyl group is introduced to the benzene ring or the aromatic ring, making a hydroxyl containing amino acid tyrosine. Okay, so phenylalanine hydroxylase enzyme needs coenzyme tetrahydrobiopterin. So deficiency of phenylalanine hydroxylase, it will give rise to a disease called phenylketonuria, because phenylalanine which will be elevated in the tissue that will go into alternate metabolic fate, giving rise to phenyl acetate, phenyl lactate, phenyl pyruvate, phenylethanolamine. So some of them are ketone derivative of phenylalanine. So they will be elevated in the tissue and altering the metabolic process in the tissue and ultimately coming out in the urine called phenylketonuria. So one of the most important phenyl ketone body is phenyl acetate, and this excess presence of phenyl acetate in the body fluids or in the urine, it will give rise to mousy or musty odor. Mousy or musty odor seen in phenylketonuria patients body fluid is because of phenyl acetate. Remember that, very important. So that's how phenylalanine is converted to tyrosine. So during this process, BH4 is converted to BH2, and this BH2 like BH4 converted to BH2, and BH2 has to be converted back into BH4, and that will be done by enzyme tetrahydrobiopterin reductase enzyme. So dihydrobiopterin is converted into tetrahydrobiopterin. So I'm sorry, enzyme there is dihydropteridine reductase, not tetrahydrobiopterin reductase. It is dihydropteridine reductase enzyme which will convert BH2 into BH4. So if there is a defect in the gene coding for dihydropteridine reductase enzyme, that leads to decreased availability of BH4, and that can also lead to elevation of phenylalanine, giving rise to phenylketonuria. And that type of phenylketonuria we call it as type 2 phenylketonuria, which is sometimes referred as malignant hyperphenylalaninemia. Why it is called as malignant hyperphenylalaninemia? Because of deficiency of dihydropteridine reductase, because it leads to decreased BH4, and this BH4 is not only necessary for conversion of phenylalanine into tyrosine, it is also necessary for conversion of tyrosine into DOPA, dihydroxyphenylalanine, in dopamine, norepinephrine, epinephrine formation. It is also necessary for the conversion of tryptophan into 5-hydroxytryptophan by tryptophan hydroxylase enzyme. 5-hydroxytryptophan further it gives rise to serotonin, melatonin, all these neurotransmitters, and also nitric oxide synthase from arginine into nitric oxide. So there also you need tetrahydrobiopterin. So deficiency of tetrahydrobiopterin will give rise to decrease in dopamine, norepinephrine, epinephrine, serotonin, melatonin and nitric oxide. All these neurotransmitters are decreased, and that will be a severe condition of phenylketonuria called malignant hyperphenylalanemia. That's because of deficiency of dihydropteridine reductase, and also it can be type 3 phenylketonuria can be because of decrease in the synthesis of tetrahydrobiopterin, a new molecule of tetrahydrobiopterin that is coming from GTP. Guanosine triphosphate is the source of tetrahydrobiopterin. So there are several enzymes involved in that. If there is any enzyme defective in that conversion process, that can also lead to phenylketonuria. So classic phenylketonuria is because of deficiency of phenylalanine hydroxylase, where patient will have fairness because tyrosine is not converted into less tyrosine means less melanin. So relatively they are fair blonde, and then excess phenylalanine in the tissue, phenylketonuria body is mousy musty order, and importantly they will have mental retardation. Why phenylketonuria patients will have mental retardation? Because there is so much phenylalanine, it will compete with the other large neutral amino acids for LNAAT, large neutral amino acid transporter in the neurons, and most of the time phenylalanine is gaining access into the neurons, and other big amino acids like tryptophan, tyrosine and the branched chain amino acids, they don't get access to the neurons. So neuronal development will be affected in phenylketonuria patients. That's why they will have severe mental retardation. So that kind of things will be more severe in BH4 deficiency because of dihydropteridine reductase or GTP conversion into BH4. So they will give rise to type 2, type 3 phenylketonuria, referred as malignant hyperphenylalanemia. So that's about phenylalanine conversion into tyrosine, and clinical aspects I have told you. Now coming to what happens to the tyrosine. Tyrosine, of course, it can go into protein synthesis because phenylalanine into tyrosine. That's why I told you some books will mention tyrosine as a semi-essential amino acid, because we can make tyrosine from phenylalanine. As long as you take phenylalanine, you don't need tyrosine actually. Okay, so now tyrosine, other than going into protein synthesis, it can go into dopamine, norepinephrine, epinephrine formation. It can go into T3, T4, triiodothyronine, tetraiodothyronine, thyroid hormone formation. It can go into skin pigment melanin formation, or tyrosine can undergo breakdown process like catabolism into fumarate and acetoacetate. Fumarate is a ketogenic, glucogenic, acetoacetate is a ketone body. That's why tyrosine, phenylalanine, they are and tryptophan, they are glucogenic and ketogenic also. Now let me go into each one of them separately. Now this is a brief thing, like tyrosine undergoing iodination process into T3 and T4, triiodothyronine, tetraiodothyronine, that is thyroxine. So active form of thyroid hormone is T3. Now tyrosine going into DOPA by tyrosine hydroxylase enzyme, rate limiting step there. BH4 is necessary here in this step, tyrosine hydroxylase step. So DOPA, what is DOPA? So tyrosine has got hydroxyl group attached to the aromatic ring. So one more hydroxyl group is attached besides that original hydroxyl group. So that means now tyrosine has two hydroxyl groups. So it is no longer called as tyrosine now. So the phenylalanine will have two hydroxyl group, first hydroxyl group and this second. So this is the first hydroxyl group. This reaction adds second hydroxyl group. So dihydroxyphenylalanine, that's what is DOPA. DO, dihydroxyphenylalanine is the DOPA. Okay, and now this DOPA undergoes decarboxylation process called done by DOPA decarboxylase enzyme, and that needs PLP, pyridoxal phosphate. Always remember, whenever amino acid derivatives undergoes decarboxylation or amino acid is undergoing decarboxylation, making a neurotransmitter, usually that needs pyridoxal phosphate as the coenzyme. Remember that. Decarboxylation of amino acid needs pyridoxal phosphate, remember. So DOPA undergoing decarboxylation by DOPA decarboxylase, you get dopamine. This happens in dopaminergic neurons, sympathetic neurons, and basically sympathetic neurons and in adrenal medulla. Okay, so that's what is how the dopamine is made. Now this dopamine is undergoing dopamine beta oxidation reaction, where ascorbate and copper is the requirement there, and becoming norepinephrine. This happens in sympathetic neurons. So up to dopamine happens in dopaminergic neurons, further two reactions don't go on in dopaminergic neurons. So dopamine is the neurotransmitter in dopaminergic neurons, because dopamine do not undergo further reaction because they don't have any enzymes which are catalyzing further reaction. That ends there for dopaminergic neurons. Like if the dopamine is deficient in or dopaminergic neurons are not metabolism of dopaminergic neurons not going on properly, that can usually dopaminergic neurons like nigrostriatal cell substantia nigra. So if there is a defect in there, it will give rise to Parkinson's disease. So remember all that. Now dopamine beta oxidase making norepinephrine, and that needs copper. So norepinephrine sympathetic neurotransmitter. All sympathetic neurons will do this. Okay. So phenylalanine to tyrosine, tyrosine to dopa, dopa to dopamine, dopamine to norepinephrine. That's where it ends. Whereas in the adrenal medulla, what happens? In the adrenal medulla, norepinephrine is undergoing a methylation process done by SAM. SAM acts as a methyl group donor. SAM is S-adenosyl methionine derivative. S-adenosyl methionine converted to S-adenosyl homocysteine, and that is done by PNMT enzyme, that is phenylethylamine N-methyltransferase enzyme, which is converting norepinephrine into epinephrine in the adrenal medulla. And remember, when the person is in stressful situation where cortisol is synthesized in the adrenal cortex, and that cortisol is going to induce PNMT enzyme, phenylethylamine N-methyltransferase in the adrenal medulla, thereby converting more and more norepinephrine into epinephrine. So more and more epinephrine can be released. So excess cortisol can also lead to excess epinephrine, and cortisol is it can break down skeletal muscle, releasing alanine, releasing glutamine from the skeletal muscle degradation, alanine, glutamine coming and making pyruvate, and pyruvate goes all the way to glucose. So excess cortisol actually lead to increase in gluconeogenesis. And also note that excess cortisol will stimulate PNMT enzyme, induce PNMT enzyme, making more epinephrine, and that epinephrine is also glucogenic, right? Because just like cortisol, epinephrine also induces pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6 bisphosphatase, glucose 6 phosphatase, glucogenic enzymes, all of them will conduct gluconeogenesis. That's how cortisol works along with epinephrine and increase blood glucose level. So that is why in Cushing syndrome there will be glucose intolerance, or when the person is in stressful situation for a long period of time, they will in fact will have glucose intolerance, ultimately leading to diabetes mellitus, type 2 diabetes mellitus, or cortisol induced diabetes. Okay, so make a connection like that. That's how epinephrine is synthesized. So there is a special product coming from tyrosine is T3T4, dopamine, norepinephrine and epinephrine. And also remember, dopamine will have a negative effect on prolactin. So whenever there is more dopamine, so prolactin, it has got inhibitory effect on prolactin. Okay. Now what happens in malignant hyperphenylalaninemia, the non-classic phenylketonuria, phenylalanine is not converted to tyrosine, means dopamine, norepinephrine, epinephrines are decreased. That means dopamine levels, when it is decreased, its inhibitory effect on prolactin synthesis is not there. So more and more prolactin is released. So in malignant hyperphenylalaninemia, when there is decrease in dopamine, norepinephrine, epinephrine, and then serotonin, melatonin, melanin, all these things are there, there will be increase in the levels of prolactin. Remember that point you got to remember. Okay, that's how you need to connect. Now coming to the melanin formation, tyrosine in melanocytes, especially in the skin cells or wherever you see the pigment in the choroid of the eyes and all that. So what happens? So the melanocytes, especially in the skin, so when upon exposure to sunlight, there will be induction in the enzyme called tyrosinase enzyme, tyrosine hydroxylase. In fact, it is referred as tyrosinase because it do not need a tetrahydrobiopterin, but it needs copper. It's a copper dependent enzyme. Tyrosinase enzyme in the melanocyte is a copper dependent enzyme. What it does, it is going to convert tyrosine into dopa and dopa into dopaquinone, and dopaquinone undergoes multiple further downstream reaction, becoming eumelanin, pheomelanins, and eumelanins are the true melanins which have got dark brown pigmentation, and that's the melanin here. So amount of tyrosine that is going into the melanin formation will determine our skin color. Okay, and that is genetically determined because the tyrosinase enzyme expression and activity is genetically determined. So and also exposure to sunlight will increase the expression of tyrosinase enzyme gene, and more tyrosinase enzyme activity through UV light, ultraviolet light increases melanin pigmentation. So that's why tanning is like when some people go to beach for tanning process, it's because of UV light inducing basically increasing the activity of tyrosinase enzyme and all the other enzymes involved in the melanin biosynthesis. Remember that point also. Now, if the tyrosine are still excess, so tyrosine can undergo catabolism, undergoing transamination reaction, tyrosine transaminase enzyme here, becoming p-hydroxyphenyl pyruvate, that is I have written it as PHPP. Tyrosinase enzyme or transamination, it's a tyrosine transaminase enzyme. Pyridoxal phosphate is the coenzyme for all transaminases. There are 17 transaminases. All of them need pyridoxal phosphate as the coenzyme. Remember. So 4-hydroxyphenyl pyruvate coming from tyrosine will be converted to homogentisate done by 4-hydroxyphenyl pyruvate dioxygenase enzyme. And then so homogentisate undergoes oxidation becoming melyl acetoacetate done by homogentisate oxidase enzyme. Homogentisate oxidase. So deficiency of homogentisate oxidase, it will give rise to what is called as alkaptonuria. And in that condition, there will be increase in homogentisate, and this homogentisate will be converted to quinone derivative. And this quinone, it has more affinity for cartilaginous tissue, and quinone, it is a black pigment, brownish to black pigment. So wherever it is accumulated over the cartilage, there will be brownish to black pigmentation. Even homogentisate coming in the body fluids in the urine sample when it is exposed to air, so oxidation of homogentisate into quinone, that will also turn urine sample black. So basically it's a black urine disease. That's what is called as alkaptonuria. So the body parts will have a blackish pigmentation, which is called as ochronosis. Ochronosis is the generalized black pigmentation in the body wherever cartilaginous tissues are there. That's one of the important signs that you see in alkaptonuria, black urine disease, black pigmentation, remember that. And that's because of excess homogentisate which is diverted into quinone derivative, and that quinone has higher affinity for cartilaginous tissue. Okay, so now homogentisate converted into melyl acetoacetate. Melyl acetoacetate can undergo mutase reaction becoming fumaryl acetoacetate, and melyl acetoacetate and fumaryl acetoacetate, so they can also accumulate if there is a deficiency of enzyme fumaryl acetoacetate hydrolase enzyme. So the fumaryl acetoacetate hydrolase enzyme, last reaction, which will break fumaryl acetoacetate into acetoacetate, which is a ketone body, and fumarate is a glucogenic, it can go into glucose formation. So done by fumaryl acetoacetate hydrolase. Deficiency of fumaryl acetoacetate hydrolase enzyme, it will give rise to type 1 tyrosinemia, which is also called as hepatorenal syndrome. So where kidneys are affected, renal means the liver is affected, hepatic cancers can be seen, and also there will be for renal tubular defect. So and also melyl acetoacetate and fumaryl acetoacetate levels will be raised, and this increase in melyl acetoacetate, fumaryl acetoacetate, so they will become succinyl acetone, and this succinyl acetone is the toxic molecule. That's what it leads to renal damage and kidney damage. Remember, so in type 1 tyrosinemia, that is hepatorenal syndrome or Richner-Rannert syndrome, so there will be elevation of fumaryl acetoacetate, melyl acetoacetate, both of them can go towards succinyl acetone, and succinyl acetone is the toxic molecule. It leads to renal damage and kidney damage. Okay, so remember that point also. That's about breakdown of tyrosine into fumarate and acetoacetate. There are type 2 and type 3 tyrosinemias which are not really that important, so that's why I am just keeping it here. Now that's about conversion of phenylalanine to tyrosine, tyrosine into various special product including the catabolism of tyrosine. Coming to tryptophan. Tryptophan can also make some special products apart from going into protein synthesis. Like 90% of the tryptophan go into protein synthesis, around like 9% of the tryptophan can go into like niacin formation, 1% of the tryptophan going into serotonin and melatonin formation and all that, right? So there are two special products that are coming, so in fact 3 special products coming from tryptophan. One is serotonin in all serotonergic neurons, other is melatonin. Melatonin is in pineal gland or pineal gland, where melatonin is it maintains circadian rhythm, that is sleep and wake cycle or dark and light cycle. which is done by amount of melatonin upon exposure like darkness. Special product is nicotinamide, which is actually niacin. Okay, so tryptophan, 60 milligram of tryptophan will make 1 milligram of niacin, that is 60:1 the conversion rate. So how that happens? Tryptophan undergoes hydroxylation process which needs BH4 there, tetrahydrobiopterin, done by tryptophan hydroxylase to become 5-hydroxytryptophan. 5-hydroxytryptophan undergoes decarboxylation process by dopa decarboxylase enzyme, PLP is necessary there to become serotonin. This is what happens in the serotonergic neurons, and serotonin is a neurotransmitter, and every neurotransmitters will undergo breakdown process, catabolism, because they are short lived. So serotonin also undergoes breakdown to become 5-hydroxyindole acetic acid. That's a breakdown product of serotonin. I forgot to explain about epinephrine, norepinephrine, dopamine. Dopamine undergoes breakdown into homovanillic acid. Norepinephrine undergoes breakdown into vanillyl mandelic acid. Epinephrine undergoes breakdown into metanephrine initially, metanephrine into vanillyl mandelic acid. So basically epinephrine and norepinephrine, they have common end product, that is vanillyl mandelic acid, but only difference here is norepinephrine gives vanillyl mandelic acid, epinephrine initially gives metanephrine and vanillyl mandelic acid. So you will have to see if metanephrine is elevated. If the metanephrine is elevated, it is along with VMA, it is coming from epinephrine. If only VMA elevated, metanephrine is normal, then think that it is coming from norepinephrine, sympathetic neuronal tumors and all that. Adrenal medullary tumor, that is epinephrine. Okay, so this is how the catabolism of these neurotransmitters, especially epinephrine, norepinephrine, dopamine, so catalyzed by two important enzymes. One is monoamine oxidase enzyme, other is COMT, catechol O-methyl transferase enzyme, methylation reaction there. Okay, so that's about it. So we are going to measure VMA, HMA and 5-hydroxyindole acetic acid in the urine sample. So the gold standard method of estimating neurotransmitters end product is a urine sample, 24-hour urine sample collection, and measuring it generally using by chromatography technique, okay, affinity chromatography and all that. Now that's how 5-hydroxytryptophan is converted to serotonin. Serotonin is a calming neurotransmitter, and decrease in serotonin will lead to endogenous depression. That's why in people suffering from depression, one or the other way serotonin levels need to be elevated. That is done by monoamine oxidase inhibitors, whereby decreasing the conversion of serotonin into 5-hydroxyindole acetic acid, or selective serotonin reuptake inhibitors are prescribed, or even tryptophan is given, excess tryptophan is given so that some of the tryptophan is diverted into serotonin. Even 5-hydroxytryptophan is also supplemented, which is eventually converted into serotonin in the serotonergic neurons. Those are some of the pharmacological aspects that you should remember. Now what happens to the serotonin in pineal gland? In the pineal gland, serotonin undergoes acetylation and followed by methylation. Acetylation and methylation. Methyl group donor there is SAM, S-adenosyl methionine. So acetylation and followed by methylation, you get N-acetyl serotonin, and N-acetyl serotonin will become methylation process, undergoes methylation using SAM as a methyl group donor, becoming melatonin, and melatonin is a neurotransmitter or neurohormone which will maintain circadian rhythm, sleep and wake cycle. Alteration in the melatonin synthesis will lead to disturbance in the sleep. Okay, so exposure to bright light all the time, so can lead to decrease in melatonin formation coming from serotonin, so that can alter or abnormal sleep pattern can happen. Remember that. Now tryptophan 60 milligram going into 1 milligram of niacin. The only important intermediates I have written here. Tryptophan into kynurenine. Multiple steps are involved, but I just written only few important ones. Kynurenine. Kynurenine into quinolinate done by kynureninase enzyme. Kynureninase, and that needs pyridoxal phosphate as the coenzyme. Remember. So conversion of kynurenine into quinolinate needing PLP, pyridoxal phosphate, by kynureninase enzyme. So deficiency of vitamin B6 pyridoxal phosphate, active form of vitamin B6 is pyridoxal phosphate. Deficiency of that will lead to decrease in the conversion of kynurenine into quinolinate. That means it leads to decrease in vitamin B3 niacin synthesis. So deficiency of vitamin B6 will also lead to deficiency of vitamin B3, remember. And also conversion of tryptophan into niacin, it needs vitamin B2, that is riboflavin, and also it needs iron, Fe2+. So deficiency of B2, deficiency of B6, deficiency of iron can also lead to vitamin B3 deficiency niacin. So at that time, what happens to the kynurenine? When pyridoxal phosphate is deficient, so kynurenine will build up, and this kynurenine will go into xanthurenate formation, and this xanthurenate will go out of the our body into urine. So there will be excess xanthurenate in the urine, leading to excess xanthurenate in the urine sample. That's indirectly, it's an indication of pyridoxal phosphate deficiency. So vitamin B6 deficiency can be indirectly measured by measuring xanthurenate levels in the urine sample. Remember that also. That's one of the applied aspect. Anyway, so kynurenine converted into quinolinate. Quinolinate is converted into nicotinamide. That's the vitamin B3 there. Now, let me take you into methionine metabolism, another important amino acid. Now, the methionine is converted into S-adenosyl methionine by adenylation process. Adenylation where adenosine is given by ATP, S-adenosyl methionine. S-adenosyl methionine undergoing a methylation reaction. There are various reactions which needs methyl group donor as SAM. So like conversion of norepinephrine into epinephrine. So in the conversion of creatine, and in the conversion of serotonin into melatonin, you need SAM as a methyl group donor. And also nucleotide in nucleotides, DNA into methylated DNA methylation process needs SAM as a nucleotide donor, and then the phosphatidyl ethanolamine formation, you need SAM as a methyl group donor. So there are so many reactions in our body where SAM is used as a methyl group donor. So when the methylation process happens, so methyl group is donated, SAM is converted to SAH, S-adenosyl homocysteine, and now S-adenosyl homocysteine undergoes hydrolase reaction done by S-adenosyl homocysteine hydrolase enzyme, giving rise to adenosine. Adenosine is released, and remember, adenosine is an inhibitory neurotransmitter in the central nervous system. Okay, so excess adenosine may actually lead to inhibition of the neurotransmission process. Anyway, S-adenosyl homocysteine converting into homocysteine after releasing adenosine. Now this homocysteine, it is in the crossroads. What happens? Homocysteine, either it can go back into methionine formation, that's one pathway for it, or it can go down into cystothionine formation. Okay, it all depends on what is the need of the cell at that time. If the cell needs cysteine formation, because cystothionine, it breaks into alpha ketobutyrate and cysteine, so homocysteine can go down into cysteine formation, or if the cysteine is sufficient, at that time if there is a need for methionine, so homocysteine is taken into methionine formation. Now let me first explain you how homocysteine is converted into methionine. Now homocysteine converting into methionine, you need a methyl group donor there. So methylation process done by methionine synthase enzyme. So the methyl group donor or the methylation is done using methylcobalamin, vitamin B12. Methylcobalamin is one of the form of vitamin B12. So this methyl group is taken from methylcobalamin, and that's when methyl group is transferred to homocysteine to become methionine done by methionine synthase enzyme. It's a transferase class of enzyme, and methylcobalamin is coming out of the reaction as just cobalamin there. Okay, now this cobalamin has to be converted back into methylcobalamin just to maintain the amount of methylcobalamin for further conversion of homocysteine into methionine, and for that methyl group, another methyl group donor is necessary, and that is N5-methyl tetrahydrofolate, and that N5-methyl tetrahydrofolate is the most abundant storage form of tetrahydrofolate in the cells. So that is getting into this reaction where B12 is converted into methyl B12, whereas CH3-THF is converted into THF, tetrahydrofolate, just tetrahydrofolate. And that is most important because tetrahydrofolate, which is there in methyl tetrahydrofolate, N5-methyl tetrahydrofolate, and that methyl group has to be taken out and releasing tetrahydrofolate, so that free tetrahydrofolate can go into various tetrahydrofolate derivatives, and that can be N5-N10 methylene tetrahydrofolate, N10 formyl tetrahydrofolate, or formamino tetrahydrofolate. So different types of tetrahydrofolates are necessary for our metabolic reactions, and that means conversion of N5-methyl tetrahydrofolate into free tetrahydrofolate is most important and that happens only if there is sufficient cobalamin to convert that into methylcobalamin. So what happens? If there is a deficiency of cobalamin, like in vegans who omit anything that is coming from animal source is the predominant source of cobalamin for us. So vegans or anyone who has deficiency of cobalamin for various reasons, cobalamin deficiency, I have a video on that, you can watch that. So cobalamin deficiency will trap this tetrahydrofolate as methyl tetrahydrofolate inside the cell, because this reaction don't go on here. Okay, so all the tetrahydrofolate in the cell is just trapped as methyl tetrahydrofolate. So free tetrahydrofolate will be decreased, and that is what is referred as folate trap, because folate is trapped as methyl tetrahydrofolate, N5-methyl tetrahydrofolate, and that is seen in cobalamin deficiency. So in folate trap or cobalamin deficiency, there will be decrease in the conversion of homocysteine into methionine. So when there is a decrease in methionine formation, that means there will be decrease in SAM, that means decrease in methylation process, and there are so many methylation process that can be affected. Okay, and that also leads to elevation of homocysteine. So decrease in cobalamin, decrease in folate will lead to increase in homocysteine, that leads to homocysteinuria, that is homocysteinuria type 2 and type 3. Now, alright, so now I am going to explain you conversion of homocysteine into cystathionine, and that is done by enzyme cystathionine beta synthase. And this cystathionine beta synthase enzyme, it needs pyridoxal phosphate, that is a vitamin B6 active form, as the coenzyme. So here in this reaction, the serine, it is going to bring the carbons of serine. It is basically entire serine, it is condensing with homocysteine to make cystathionine. Remember, methionine is a sulfur containing amino acid, and that sulfur, it is now part of homocysteine. Now that homocysteine sulfur, it is basically entire homocysteine condensing with serine, making cystathionine, and cystathionine still has that sulfur coming from homocysteine. Okay, now this cystathionine, it breaks into alpha ketobutyrate and cysteine, and now the sulfur is there in cysteine, and the carbons of cysteine, it is actually coming from serine. Alright, so what I am saying here is cystathionine has got sulfur, and that sulfur, when the cystathionine breaks into cysteine and alpha ketobutyrate, and that is done by enzyme cystathionase enzyme, and this cystathionase enzyme, it also needs pyridoxal phosphate as the coenzyme. So what happens here? So when cystathionine breaks into alpha ketobutyrate, see the carbons, all the carbons of alpha ketobutyrate is actually coming from homocysteine, and that actually coming from methionine. So the carbons of methionine, now they are present in the form of alpha ketobutyrate. What happened to the sulfur? Sulfur of methionine, which was part of homocysteine, now it become a part of cysteine. And what about the carbons of cysteine? Carbons of cysteine actually coming from serine. Most important, you got to remember this. Now, cysteine can undergo protein synthesis, and also cysteine can have different special products that can be made. Cysteine is used in the formation of glutathione, that is GSH. Glutathione is gamma glutamyl cysteanyl glycine, which is a most important intracellular antioxidant in our cells. And also cysteine can go into taurine formation. And now you know taurine is used in the conjugation of bile acids. You know, bile acid conjugation, taurine, Colyl-CoA, shinodioxycolyl-CoA, converting into taurocolate and tauroshinodioxycolate. These are the bile acid conjugation where taurine is actually a derivative of cysteine. Make sure you remember that point. Now let me take you into alpha ketobutyrate. This alpha ketobutyrate, further it will be converted into propionyl-CoA after decarboxylation process, and this propionyl-CoA can go into gluconeogenesis. It becomes part of this Bob needs vitamin M. So I told you vitamin M, that is valine, isoleucine, threonine and methionine. So that's where is the methionine coming in there. Okay, so that's what happens to metabolism of methionine into homocysteine into cystathionine, alpha ketobutyrate, propionyl-CoA, and going into glucose formation and also cysteine formation. And also whenever there is high levels of cysteine, this cysteine will have a negative effect, feedback negative effect on cystathionine beta synthase enzyme. Remember that. Okay, so that's about methionine metabolism. So if there is a defect in cystathionine beta synthase, that will give rise to type 1 homocysteinuria. Signs and symptoms of homocysteinuria is similar to Marfan syndrome, but there are some additional signs. What are the additional signs other than arachnodactyly, spider finger and all that? Other than that, patients with homocystinuria type 1, so they will have mental retardation, they will have osteoporosis, they will have deep vein thrombosis, and they will have lens dislocation, which is in infero-medial direction rather than supero-temporal direction. So you got to remember all that. So that's about, and that's type 1 homocystinuria because of cystathionine beta synthase enzyme, which has mutated, and the type of mutation is where the enzyme has decreased its affinity for pyridoxal phosphate. That means when pyridoxal phosphate or the pyridoxine is given, this enzyme activity can be improved. That's why pyridoxine is the treatment of choice for homocystinuria type 1. Now coming to type 2 and type 3 homocystinuria. So type 2 and type 3 homocystinuria, so it is because of deficiency of vitamin B12 or deficiency of folate. So folate deficiency or cobalamin deficiency can lead to type 2 and type 3 homocystinuria. So that's about it there. So the difference between type 1 and type 2, type 3 homocystinuria is in type 1 homocystinuria, there will be elevation of homocysteine along with the methionine, whereas in type 2 and type 3, there is elevation of only homocysteine but not methionine. So remember that. Now coming to branched chain amino acid metabolism. So the branched chain amino acids, we have valine, isoleucine and leucine. These are the three branched chain amino acids. So these branched chain amino acids, when they undergo catabolism, especially because branched chain amino acids can go into protein synthesis, especially skeletal muscles are the major consumers of branched chain amino acids because skeletal muscle proteins, they need more branched chain amino acids. Anyway, so when valine, isoleucine, leucine undergoes catabolism, so all of them will undergo similar first reaction, that is transamination reaction, where alpha ketoglutarate is converted to glutamate. Okay, and the amino group of valine, isoleucine, leucine given to alpha ketoglutarate, where they become part of glutamate. The carbon skeleton of valine, leucine, isoleucine, they will become alpha keto isovalerate, alpha keto beta methylvalerate and alpha keto isocaproate. These are the keto acid derivatives of valine, isoleucine and leucine. So what happens to these branched chain keto acids? Branched chain keto acids, they will undergo reaction catalyzed by branched chain alpha keto acid dehydrogenase enzyme complex. Remember, this branched chain alpha keto acid dehydrogenase enzyme is very similar to alpha ketoglutarate dehydrogenase enzyme complex here, and also pyruvate dehydrogenase enzyme complex. That means this enzyme, just like other two enzymes, it has E1, E2 and E3 subunit. It needs 5 coenzymes. What are those 5 coenzymes? Thymine pyrophosphate, lipoic acid, pantothenic acid, riboflavin and niacin. These are the 5 coenzymes that are necessary for these 3 enzymes which contains E1, E2, E3. So deficiency of branched chain alpha keto acid dehydrogenase enzyme complex, it will lead to elevation of all these branched chain alpha keto acids, that is alpha keto isovalerate, alpha keto beta methylvalerate, alpha keto isocaprate. They are all elevated in the blood, and since this is a reversible transamination reaction here, so they will also undergo transamination process back into valine, isoleucine and valine, so they also will be elevated. So deficiency or defect in branched chain alpha keto acid dehydrogenase enzyme lead to elevation of branched chain alpha keto acids along with elevation of branched chain amino acids. So elevated branched chain alpha keto acids, they will lead to a special odor to a body fluids, and that is a burnt sugar or a maple syrup sugar odor. Okay, it's the burnt sugar odor. Remember, body fluids will have a burnt sugar odor, and that's why the name of the disease, because of deficiency of branched chain alpha keto acid dehydrogenase deficiency, is called as maple syrup urine disease. Okay, so body fluids will have a maple syrup odor or the burnt sugar odor. Remember that. So, and also patients with branched chain alpha keto acid dehydrogenase deficiency will have a metabolic acidosis and severe complications including mental retardation. Usually it's a severe condition and survival will be difficult. So otherwise what happens? Alpha keto acids of branched chain amino acids, they will all become their corresponding CoA forms, that is isobutyryl-CoA, methylbutyryl-CoA and also isovalyryl-CoA. And furthermore, they will undergo catabolism making propionyl-CoA. As you see, valine is making a propionyl-CoA and a propionyl-CoA glucogenic, and that's why I have written here. Bob needs vitamin M, valine needs, it will give propionyl-CoA. And then look at isoleucine. Isoleucine gives propionyl-CoA and acetyl-CoA. Acetyl-CoA can become acetoacetate, which is a ketone bodies. That means it's a glucogenic and ketogenic. So isoleucine is also part of Bob needs vitamin M. Now leucine, let's look at leucine. Leucine will become means it will give acetoacetate, which is a ketone body, and acetyl-CoA, that also become acetoacetate. Basically it's a ketone body. So leucine is a purely ketogenic amino acids. Okay, so that's what is the metabolism of valine, isoleucine and leucine. So this covers important amino acids with clinical applications related with that. So you can look into glycine metabolism. So glycine metabolic defect is primary hyperoxaluria is there. Serine metabolism, glycine metabolism, especially in connection with tetrahydrofolate derivative, you can look into that. So basically I have covered tyrosine, phenylalanine, tryptophan, methionine and cysteine, and then I have covered valine, leucine, isoleucine. Alright, so look at this succinyl-CoA, one of the TCA cycle intermediate, because TCA cycle it's an open cycle. It is also called as amphibolic cycle, because some of the things in TCA cycle can be taken out to go into some other metabolic process, like alpha ketoglutarate can go into glutamate formation, further into GABA formation, or oxaloacetate can go into aspartate formation, or malate go into gluconeogenesis, like that. Succinyl-CoA here, succinyl-CoA can be taken out of TCA cycle, especially in the matrix of mitochondria in all erythropoietic cells or any cell that needs bilirubin, any cell that needs heme for heme containing proteins like electron transport chain and all that. Succinyl-CoA condensing with glycine to make delta amino levulinic acid. This is done by delta amino levulinate synthase enzyme. Now, 2 delta amino levulinic acid combines with one another to make parfobilinogen, and parfobilinogen becoming hydroxymethylbilane by HMB synthase enzyme. Hydroxymethylbilane converted into uroporphyrinogen 3, coproporphyrinogen 3, protoporphyrin 9, protoporphyrin 9, and ultimately it will combine with ferrocyanide to become heme. And this is how succinyl-CoA can be diverted into heme synthesis. And this happens in the erythropoietic cell and also in cells that needs heme as a part of the protein, like heme protein, which is like tryptophan, pyruvate, catalase enzymes. So electron transport chain, cytochrome oxidase, they are all heme containing proteins. So they need succinyl-CoA, which is coming out of TCA cycle to go into heme synthesis. So defect in any of these enzymes involved in heme synthesis, they will give rise to different types of porphyria, and the most common type of porphyria is Porphyria cutanea tarda, because of urodecarboxylase, uroporphyrinogen decarboxylase enzyme. And the second most important porphyria is acute intermittent porphyria. So different types of porphyrias are there. You may go over the details of those porphyria. And also heme synthesis can be affected by lead poison, because dehydratase enzyme and ferrochelatase enzyme, last enzyme, are lead sensitive enzyme. So that can make that can basically inhibit heme synthesis. Now once heme is made, heme can combine with globin to make hemoglobin, or heme become part of a protein, and when that protein undergoes degradation, heme can also undergo degradation. So heme can break into biliverdin. So heme oxygenase will release carbon monoxide while it converts heme into biliverdin. Okay, and this is the only reaction in our body which gives carbon monoxide as the byproduct. Okay, so that means the extent of hemolysis that is going on in our body can be measured by the amount of carbon monoxide that is synthesized. Okay, so once you get biliverdin, biliverdin is taken into the liver, biliverdin is converted to bilirubin by biliverdin reductase enzyme, and this bilirubin is water insoluble. This water insoluble bilirubin is taken into the liver, where it will undergo conjugation process with UDP glucuronate, because I told you here UDP glucose converted to UDP glucuronate, and same UDP glucuronate can be used in conjugation of bilirubin into bilirubin monoglucuronate, and then one more UDP glucuronate added becoming bilirubin diglucuronate, and that bilirubin diglucuronate is water soluble molecule. It will be secreted into the biliary canaliculi going into the duodenum, and in the duodenum bilirubin diglucuronate undergoes unconjugation of that glucuronate releasing urobilinogen, and urobilinogen, most of it will be secreted in the feces. Only less than 3 milligram of urobilinogen you can find in the urine sample. So that's a bilirubin degradation, heme degradation process, making bilirubin conjugated, unconjugated process and all. Defect in any of this process can lead to hyperbilirubinemia and jaundice. There are different types of jaundice, pre-hepatic, hepatic and post-hepatic jaundice. You can watch my video on this topic, it is there in YouTube, just in case if you didn't get that concept. So that's about heme synthesis and heme degradation. So with this I have covered most of the important metabolic pathways that you come across, and also the most of the important enzymes, clinical aspects I have covered it in brief. So that's about it. I hope this particular review video will really help you in reviewing everything in a short period of time, and this is most important for you to remember as a quick review for your exam. So if you didn't get it first time, you watch it repeatedly, so maybe you will get better when you watch second time. And also if you have any question, any doubt in some of the individual concepts that I have taught, so I have videos on those individual concepts also. You can go over those videos separately, understand them and come back to this and continue. So overall, if you understand whatever I have explained today, that covers 80% of the biochemistry. That means safely you can go over and take the exams. At least you are sure about 80% of the performance there. Again, so to improve your performance, you will have to go into in depth, in detail, which is unfortunately I cannot do that all that in a short period of time. You can go over my detailed individual videos in my channel. So that's about it, and I hope it really helped you in understanding or like reviewing this entire metabolic pathways. I'll see you in my next video, and until then you take care. And if you have any questions, so let me know in the comment section below. And if you have any difficulty in understanding, let me know. I can make a special video on that. And also if you don't find some of the individual concepts in my channel, so let me know. I'm going to make it for you. Till then, you take care.